because our life is very easy according to plastic products. But in this plastic, there are a lot of chemical compounds like bisphenol A, phthalates, azo dyes, heavy metals, and different compounds too. And I will explain these chemical compounds. So these items really try to reduce the production of growth hormone, estrogen and testosterone hormone, and in the future, obesity, diabetes, asthma, cardiovascular disease, and liver damage. It increases the risk of breast and prostate cancer. Hi again, can you hear me?
Yes, sir, we can hear you.
Yes. Okay. Uh, Continue again because yes. Until 4 and 20 years ago, yogurt was sold in glass or clay containers, water in glass, demi-johns and iron and milk in glass bottle. Very commonly iron, but in old time we can use this glass bottle, but unfortunately nowadays everywhere, every time we can use plastic bottle. Yes, so before I want to explain our study because in the first time we studied about a plastic bottle into water, and unfortunately we found 5 chemical compounds into plastic bottle water, unfortunately. And I used LCMS IT TOF device. Really, these chemical compounds, chimasorb 81, and these olamide and ergaphos, like these chemical compounds. After we studied about sunglasses too. So I found two chemical compounds too, antioxidant and BHT. So really, these chemical compounds damage our body. If you want, you can read later, of course, because I have little time, I know. But really, in the future, these chemical compounds pass our body, where I am sure one day these chemical compounds affect our body. And in addition, in the world, everywhere there are a lot of different cancer types, unfortunately. So plastic plates, spoon, fork, very important to in the world. Everyone use this plastic plates, spoon, forks too, because maybe we need to nowadays, according to coronavirus, but really when we use these plastic things, soluble and in the future intervened by the Ministry of Health. Plastic cups too. A beverage served at 70 and 90 degrees in tin. Cost-effective plastic cups can dissolve the polymer, releasing careful carcinogenic monomers into the drink. Fatigue, irritability, difficulty sleeping, low platelet count, and lymphatic abnormalities may occur due to polystyrene in contains, really. Foam cup, like this, very important, and really this releases monomeric groups, potential introduction, the toxic substance into the body, thought ingested. Paper cups too, really very important. Maybe everyone use these plastic cups, but this chemical consists on surface as paper cups penetrate into our body. When used for tea, coffee, and similar hot drinks, this situation invites cancer. So a doctor from Indian Institute, Goel, a person who drink 3 cups of tea or coffee, please again read, a day from a paper cup will ingest 65,000 teeny microplastic particles that cannot be seen with the naked eye. Very
Dr.
important.
So sorry.
Yes.
Can you open your presentation? We can't see presentation. So sorry.
And now, yes, again. Yes, can you see
Yes.
now?
Now, yes. Yes.
Yes,
Now, yes.
okay. Okay, sometimes stop, I think. So tea bags again, because the bags porous polymer-based structure, potential temperature-induced charge, and use of metal stable and are unstable if made from synthetic fibers or polymers. The material could harm health, particularly affecting the liver and kidneys too. So really, maybe in old time I used the tea bags in my office, but now anymore I didn't use tea bags because tea bags should be made of natural fibrous material that resist health. Synthetic cellular fibers should be avoided too. Because microplastic have been in our tea bags and cups for years. My Yale University chemical engineering professor Natalie studied plastic tea bags and found that steeping them at 95 centigrade degrees released 11.6 billion microplastic and 3.1 billion nanoplastic into a single cup. Please think, if we drink tea bags, we can take a lot of microplastic together too. So a very important study is to non-returnable plastic bottles and containers are made of polyethylene and polyvinyl chloride, while returnable containers are typical made of polycarbonate too. Harvard University, very important studies, studied data on nearly 5,000 children age 9 and 14 and found that girl who consumed more than 1 glass of sugary carbonates drinks daily entered puberty an average of 2% 8 months earlier than those who drank them 2 or more times a week. Nowadays, our problem really. And treating our oceans and seas microplastic too, because microplastics smaller than 5 millimeters break off from largest plastic pieces and oceans and seas. They appear in products like shower gels, toothpaste, and makeup mixing with water. Some dissolve in water while others remains as particles. Whether they dissolve or not poses a threat to both an ocean and marine life. Yes, sometimes I look your page too. Yes, continue. And microplastic coming to our dinner tables. Because I think people likes salt. So I am sure in the salt there are a lot of microplastic into salt too. And before I wrote my newspaper, so nowadays I am sure we drink plastic laced water flowing from our taps. It's really very, very important, really. And there is a very important manuscript too, because the Guardian reported microplastic were found in the placentas of for healthy pregnant women. Again, please, placentas of our healthy pregnant women, affecting both the mother and fetus as well as the fetal development membrane too. And microplastic pollution is worsening daily with plastic in water and food, even affecting babies in the womb. Italian scientists who previously discovered microplastics in the womb have now detected them in breast milk, unfortunately. They warn expected mothers to avoid all types of plastic production. Again, scientists detected hundreds of microplastics in human feces. And microplastic detected in human blood for the first time. Human blood in this, yes, really. And very important, microplastic were detected in the lungs of liver humus for the first time. So when we take air, so we can take air, oxygen, the other gases, and together microplastic too. So our lungs, there are a lot of different microplastic pieces. And warning from scientists, microplastic detected in clothes. Again, think clothes. In clothes, there are a lot of there are different microplastic sizes too, really. And microplastic detected for the first time in freshly fallen snow in Antarctica. In Antarctica, there is little microplastic sizes. And very important again studies too, this microplastic brain in the brain may have the power to change our behavior. So this microplastic in my conference generally I says affect dementia patients, Parkinson and Alzheimer really this effect and in the world increase these dementia patients, Alzheimer disease, Parkinson disease too. And every week we swallow a credit card size amount of plastic. So where does this plastic go? Very, very important. Islam explains the complex anatomy of the airway and the flow behavior in the nasal cavity and chromexis cause microplastic to deviate from the flow path and accumulate flow velocity, particle inertia, and asymmetric anatomy. Increase the concentration of accumulation in these areas. Plastic bags, very important too, are widely used to their lightness, low cost, and convenience. The plastic industry is rapidly growing with rising imports in Turkey. Despite risk like surgeons, we commonly use plastic bags for various items, including freshly baked bread. Millions of kilograms of plastic bags are thrown into the oceans every year. One of the main reasons the Earth's surface doesn't turn into a plastic dumps into is the use of ocean floor as a landfill. Plastic bags undergo chemical decomposition under sunlight. Okay, yes. Uh, Plastic bags undergo chemical decomposition under sunlight over time. They break down into smaller and more toxic petropolymers. As a result, our soil and water get poisoned. Ultimately, these microplastic toxic particles enter the food chain. That is when the future of nature life is at risk. Yes, we may. This very important too because we see in TV programs every time, and we found different diet animals of diets because we may unknowingly become the cause of death for one or more animals due to the waste we scatter in the environment. The dispersal of plastic bags threatens the lives of over 200 marine creatures, including mammals like whales, dolphins, and seals, as well as fish and sea turtles. And really, maybe you can read too, because a family was excited to see a bird building a nest in front of their window and watch it daily. Unfortunately, they noticed that the bird was collecting rusty nails, wires, and plastics for the nest. It seems the bird gathered these from the nearby construction site. Due to these harmful materials, the bird couldn't keep the nest warm and eventually abandoned it, leaving the eggs behind. So dangerous gases are released directly disposal of plastic bag. They clog, savory, and cause them to overflow. And we finished, I think. These bags black bag very problem too because there are a lot of different dirty bacterials and chemical compounds too because this picture very important. Black bags can sometimes contain herbicide and insecticides as they may be made from containers that previously held these chemicals too. And we can use instead of plastic bag because really in the world travel long distance appearing from the Arctic seas to Falkland Islands, European and replaced plastic baked paper bags maybe we can use. And really this plastic pollution affect climate change too because in plastic there is carbon atoms and these carbon atoms may be carbon dioxide and this carbon dioxide climate change too. So what should we do? Simple supply. We have to use glass in our kitchen, for example, and glass. Of course, we have to prefer continue. Really, this presentation is very important because this presentation is interested with our health life. So again, thank you for your participation. And maybe because I have a little time, again, thank you very much.
Thank you so much, Dr. Hamdi. It was really a very elucidative and a very informative presentation. It had a lot of awareness regarding the microplastics and how they're infesting our lives and how they are infested in our environment as well. And you are really very right that it is related to our health and it is related to our lives. So I am honored to have you as our presenter, and we look forward in collaborating with you in future also. And moving forward to the next presentation, I would like to call Dr. Malik Ziari. She is from Ibn Tufayl University, Morocco. Over to you, ma'am.
Hello, everyone. Thank you. I will share with you my presentation. I hope you can hear me.
Yeah, you are clearly audible.
Okay, that's great. Can you see the slides?
Yes, we can.
Okay. So, hello, everyone. I'm Malak Zirari, a researcher in analytical and environmental chemistry at Ibn Tufayl University in Morocco. Today, I'm excited to share our research on the nutritional composition, phytochemicals, and antioxidant activities of Avis marocana needles. This unique plant holds great potential not only for supporting human health, but also for contributing to environmental sustainability. As we move toward a more sustainable world, it's essential to explore different plant sources for nutrition. Plants offer more than just food. They also provide various health benefits. Avis marocana, a fierce species found only in Morocco, is one example. Today, I'll share our research on the unique benefits found in the needles for this rare plant. Now let's discuss why we choose to focus on Avis marocana for our research. Avis marocana is found exclusively in Morocco's Rif Mountains, which makes it not only rare but also of significant interest due to its unique chemical profile. Other types of Avis trees have been studied for their health benefits. For example, silver fir has shown anti-inflammatory and antioxidant effects, while Korean fir has shown potential for reducing oxidative stress due to its high level of polyphenols. Based on these findings, we hypothesized that Avis marocana might have unique benefits due to its special habitat and chemical profile. This inspired us to study the specific properties of Avis marocana needles in our laboratory. Our research aimed to evaluate the nutritional composition of Avis marocana needles, analyze their bioactive components, and assess their antioxidant activities. By investigating these aspects, we hope to uncover the potential application of this plant in promoting health and wellness. To achieve our objectives, we started by analyzing the nutritional content of Avis marocana needles using AUAC methods and assessed mineral content with ICPOES. In the second step, we extracted the chemical compound using substrate extraction with methanol as the solvent. For further chemical analysis, we used gas chromatography, mass spectrometry, GCMS, to identify and quantify specific compounds. Finally, we assessed antioxidant activity, including a DPPH assay to evaluate the extract's ability to neutralize free radicals. Together, these methods provide a detailed view of the nutritional and bioactive potential of Avis marocana. Now let's discuss our findings regarding the nutritional composition. Our proximate analysis revealed that Avis marocana needles are rich in essential nutrients. We found significant level of carbohydrates, crude protein, crude fibers, crude fats, ash, and moisture. Moreover, we confirmed that the presence of crucial nutrients such as chlorophyll, carotenoids, and vitamin E, which highlights the needle's complex nutritional profile. These findings suggest that Avis marocana needle can contribute positively to dietary needs. Minerals are essential for our health and play a big role in keeping both our body and mind strong. They are needed for many functions in our body, like keeping our bones healthy, supporting muscles, and boosting our immune system. In our study of Avis marocana needles, we discovered that they are rich in important minerals. This includes key minerals like potassium, sodium, calcium, magnesium, and phosphorus, which support our energy and bone health. The needles also contains iron, zinc, and manganese, which help with immune function and protect our cells. This mineral-rich profiles shows that Abies marocana needles might be a valuable addition to a healthy diet, offering potential health benefits. Our extraction process achieved a yield of 27.467%. This means we successfully extracted a valuable portion of the compound present in the needle powder. Next, let's take a look at the phenolic compound we found. These include polyphenols, tannins, and flavonoids, each known for powerful bioactive properties. Polyphenols, which we found in the highest amount, are essentially valuable for their antioxidant benefits. These compounds are widely studied for their potential in health and environment application. In short, Abies marocana could be a valuable resource not just for its chemical profile, but for what it can contribute to fields like pharmaceuticals, nutrition, and sustainable industries. This is just the beginning of what we can explore with this incredible species. In our research, we analyzed the needles of Abies marocana and found several important components. The methanolic extracts contain ketones, alcohols, fatty acids, phenols, glycerides, and phytosterols. The main compounds are fatty acids and phenolic compounds, especially polyphenols. Fatty acids are essential for building our cells, tissues, and organs, and they are also involved in forming compounds that support our health. Studies even suggest that omega-3 fatty acids may help prevent certain types of cancer. Besides, polyphenols are known for their antioxidant properties. They help protect our cells from damage and support heart health, reduce inflammation, and may even help prevent cancer. Altogether, these beneficial compounds suggest that Abies marocana needles could be a valuable addition to our diet with promising health benefits. One of the most exciting results from our research was discovering the strong antioxidant power in the methanolic extract of Abies marocana needles. Antioxidants are essential because they help protect our cells from damage caused by free radicals. This damage, called oxidative stress, is linked to chronic disease like heart disease, cancer, and diabetes. Using a method called the DPPH kidney assay, we found that the needle showed high level of free radical inhibition. In simple terms, these needles can effectively neutralize harmful molecules in the body, potentially lowering the risk of certain disease. What's even more exciting is that these antioxidant properties could make Abies marocana useful in food and health products. This research really highlights the potential of Abies marocana as a natural beneficial resource. To sum up, our study shows that Abies marocana needles have a great nutritional and health potential. Adding them to our diet could benefit both people and the environment. Looking forward, we see significant opportunity to develop innovative products for the food and pharmaceutical and cosmetic industries. These products can support green chemistry and create economic opportunities for local communities. Our next step is to conduct clinical studies to explore the full therapeutic potential of these compounds, particularly to targeted health applications. Thank you for your attention.
Thank you so much, Dr. Malik Sarai, for such an informative presentation. We all loved it. so much. So
Thank you.
without wasting, thank you so much for joining us. I think you have joined us for the very first time, and we are so honored to have you on our platform. And all right, without wasting another minute, I am going to call upon this virtual stage our next presenter, that is none other than Dr. Kamala Badalov, and she is from Azerbaijan Medical University. She has joined us in many of our online webinars and international conferences, and we are so happy to have her once again with us. Over to you, Doctor.
Okay, first, welcome to our conference as our guests. So thank you for your invitation, my dear friends. I see here a lot of my friends, for example, Hamdi, Tamel, and other friends. Thank you very much again. And now, if possible, I would like to present my presentation. I will share. Yes. Sorry, can you see presentation or not?
Yes, ma'am, we can see your presentation.
Okay, very good. So I would like to present my topic, determination of the titanium dioxide, metal nanoparticles, titanium dioxide and silver nanoparticles in living organism, and in our work we used human urine. So outline plan was introduction, determination of nanoparticles, objective, sample preparation, instrumentation and the experimental part, results, discussion, and also compilation. And in introduction, we will use application of nanoparticles in the several areas. About we will use toxicity, risk, hazard, and exposure of the nanoparticles. So you know that titanium dioxide nanoparticles we can apply, we can use in food industries, in cosmetic industries like toothpaste and sunscreen, and also in bakery industries. Then we can apply silver nanoparticles as antiviral, as antifungal, as anti-inflammatory, as antibacterial in cosmetics, in human health care, water treatment, in drug carrier, textiles, imaging, and in other several areas. So why we decided to determine titanium dioxide nanoparticles in human urine? Why this compound is toxic compound? And also that titanium dioxide titanium dioxide nanoparticles can translocate from the initial gates to entry and accumulate in them. And titanium nanoparticles accumulation in the brain cause brain damage and neurotoxicity. Titanium nanoparticles also exposure results in microglia activation, ROS protection, and inflammation. So ROS, as you know, the reactive oxygen species production induced genotoxicity and cell damage. So international agents for the research on cancer classifies titanium dioxide as a group 2B carcinogen, which means it is possible carcinogen to humans. And this was based on an animal study showing inhaling high concentrations of titanium dioxide dosed may lead to lung cancer. So in this slide, I showed for you some examples, but for example, one of them, inhalation aerosols of 8.6 milligram to meter titanium dioxide for the 7 gram per day. It was for up to 1 year. And the type of number of animals were 24 female with rats. And as a result, lung burden, titanium dioxide nanoparticles was found in the macrophage and in the perivascular spaces. And as examples, as you see from the slide, the next slide also. So the regulation which you can see in slide, published in official journal of the European Union on 14th July, amends Annex 6 to cosmetics regulation and to introduce the export. It is nanoform and make it an ultraviolet filter except in spray products. So risk hazard and exposure. Hazard-driven approach, this one, indication of silver nanoparticles like ecotoxicity, like bacteria, Daphnium magna, zebrafish, fish, human, and exposure-driven approach like silver nanoparticles released from textile paints, washing machine, water treatment, and other. And why are silver nanoparticles they are safe or not? European Commission and it is non-food scientific committee on emerging and the newly identified health risk published is a final opinion on nanosilver safety, health, and the environmental effects and the role in antimicrobial resistance. While it is more and more widely used in consumer products, including cosmetics, experts noted serious knowledge gap and the need for further research. The Commission published as explanation. So in this slide, you can see some diseases associated to nanoparticles exposure, where, for example, our nanoparticles can affect to brain with shown Parkinson's disease, Alzheimer's disease, and other. In the lungs, asthma, bronchitis, cancer, and in several organs they can affect. So our objective was main goal in our research was determination of metal nanoparticles in urine. For this, we have performed the steps as follows: optimization of a method of quantification of nanoparticles in samples of urine by ICP-MS. This is our experimental part. You can see some devices which we used in our research. So how we prepared our samples for titanium nanoparticles, we used urine. Then we mixed it with glycerol up to 1 to 15 we diluted with glycerol. Then for the silver nanoparticles, urine we diluted with glycerol, 1% solution of glycerol up to 1 to 10. And during analysis for titanium, we can see which you used urine dilution 1 to up 15, and as it was before centrifugation, and we used the urine diluted 1 to 15 after centrifugation, and also urine diluted 1 to 100 before centrifugation and after centrifugation. And we use some CD standard deviation, concentration part, which you can see from our research, and most regular size in our device. So this one from our research, for example, in dilution 1 to 15, before centrifugation and after centrifugation. For example, without centrifugation, you can see that the number of the nanoparticles, they are higher than the with centrifugation. As you know that why we need in centrifugation to separate urine from the proteins. And this one showed this size histogram showed the number of nanoparticles before centrifugation and after centrifugation. Then this one is precision of the titanium dioxide nanoparticles, where you can see some mass flux, some counts. Then other side histogram taken from device of ICP-MS. This one, titanium nanoparticles in our 11 samples, in 11 urine samples. Then about the calibration curve, where we use concentration, mass flux, and other one. So size histogram from the titanium dioxide nanoparticles. And accurate precision for the silver nanoparticles. Some size histogram for the silver nanoparticles again, and precision for the silver nanoparticles in 11 urine samples, where we use, for example, ECT, RST, ionization efficiency. Then we used concentration part, dissociation concentration, and also most frequent signs, which you can see from the table. And their calibration curves taken from our device during research. The size histogram for the silver nanoparticles. Analytical recovery. We use analytical recovery for the silver, for titanium nanoparticles, and we used three different sizes for our nanoparticles, with 20 nanometer, with 14 nanometer, and the 16 nanometer. First, we analyze this just urine. Then we analyze this urine and plus 20 nanometer of silver nanoparticles, urine plus 14 nanoparticles of silver again, and with the diluted urine with 16 nanometer of the silver nanoparticles. And after it, we analyze separately our nanoparticles in different sizes. And our results, which were taken from our device, ICP-MS. So we calculated sensitivity, their load and log parameters. Then again, our samples for the analyte for the silver, where we used glycerol and load log parameters. So also sensitivity for load log parameters is dissociation concentration, load in size, and also load in size. But after using ICP-MS device, also we tried to use other device as synthesizer Nano ZS90, affordable molecular particle size and zeta potential analyzer, an entry-level system for the measurement of particle size and molecular size at a 90-degree scattering angle using dynamic light scattering. Also with the ability to measure zeta potential and electrophoretic mobility using laser Doppler. So these high size distribution by intensity, like histogram, were taken from this device. But after it, we decided to use electronic microscope TEM and SEM images of these urines, which were taken from the 11 or 12 volunteers of our university. So after this process, we used this, for example, there are some images of how the adults, first one, same image, scanning electron microscope image of how the adults, woman 37 ages, is also urine. Then other one, same image of how the adults, again, woman 37 ages. Then men 48 ages. This one is 10 images. Again, same image of how the adult men 45 ages. As you see, we use here 50 nanometers. And the bigger nanoparticles which was found in the urine. So, same image also men 45 ages urine. And same image of how the adults, woman 25 ages urine. As you see, the size and the shape nanoparticles, they are different in this image, this electronic microscope image. So, thank you very much for your attention. Shukran and teşekkür ederim. If you have any question, you can ask. And at last, I will show you our Getter team. They're from Spain. We're taken in Spain in Santiago de Compostela University.
And your presentation was in depth and so clarifying, and it was both educational and inspiring. Thank you so much for joining us, and we look forward to have a lot more collaborations with you in future. If you have any questions, dear audience, if you have any questions, I would like you to please drop those questions into the chat box. The next speaker, we have Dr. Ibrar Hussain with us from ICCBS, University of Karachi, Pakistan. We hand over the virtual stage to you, sir.
Yeah, assalamu alaikum. Am I audible?
Walaikum assalam. Yes, sir, you are clearly audible.
Okay, let me share my screen.
And it's a humble request to all the speakers that after you present your topic, please do not forget to stop sharing your screen, as it will create a technical issue for the upcoming speakers.
Is my screen visible?
Yes, sir, your screen is visible. Thank you.
Okay, thank you. So let's start. Bismillah ar-Rahman ar-Rahim. First of all, I want to say thank you for the organizing committee and all the participants that are present virtually to International Conference on Advances in Life Sciences. I am Varar Hussain, pursuing my PhD as a research fellow from International Center for Chemical Biological Sciences in the field of probiotics. So today we'll just talk about a new horizon field which is called probiotic as medicine. Can we use probiotic as medicine? Because I'm working from the last 3 years on probiotics. These are the object of my presentation, which include the understanding of probiotics therapeutic potential, the properties of the probiotics and their concepts, and how probiotics is used as medicine. What are the diseases which are treated with the help of probiotics and their advantages and disadvantages? So, following the contents of this presentation include a brief overview about the probiotics, history, selection, properties, and benefits, and then probiotic as medicine, their properties, guideline, and precautions, what are disease treatment, and their example, and finally the summary. So let's start from very beginning that once we are talking about any microorganism, so our mind just goes that all microorganisms must be pathogenic. So I want to clarify your concept that all microorganisms are not pathogenic. These some of them are very beneficial, and this slide just showed the various beneficial aspect of microorganism, particularly the biological and medical research and those which are involved in the treatment metabolic disorders. So generally, microorganism, approximately 159,000 different species of microorganisms are identified so far, and these are present and ubiquitously with varying number of species and genera in different environments. If you're talking about particularly the humans, so In human, these microorganisms can contribute up to 3 million microbial gene, which is about 150 times more than the human genome. If we are talking about the weight, so they can contribute up to 2 kilograms to the human weight, and this just show that how much the microorganisms reside inside our body. I just want to clarify one thing more, which is microbiome or microbiota is a term which are used for the collection of microorganisms which are associated with particular items or substances, or for example, particular organ, and if you're talking about the human. So among the all other microbiome, the gut microbiota is crucial important because they can contribute and they can influence all the physiological function of the organism, host organism. About one third of the gut microbiota is similar to all individual, but two-thirds of them are different, which can affect the different aspect or different properties of the individual. So among the sea of microorganisms, some microorganisms are called good bacteria or friendly bacteria. This concept was initially introduced by a Russian Nobel laureate scientist Elementenkov in 1907, and he proposed the theory of longevity, which proposed that if we can change the world, we can change the gut microbiota with friendly bacteria, so they can increase the longevity, can increase the lifespan of the host organism. So this concept just followed by other researchers around the globe, and they can enhance and can explain by the World Health Organization in 2002, and then finally propose a proper definition by International Scientific Association for probiotics and prebiotics in 2013. And they define probiotics are live microorganism when consumed in adequate amounts, they can confer a health benefits on the host. So clearly indicate that probiotics are microorganisms which are live in nature, which have some beneficial properties and must be taken in some specific amount. This slide just indicate from our published material that how the concept of probiotics from the major developments occur from the concept of longevity to the vast health benefits of the probiotics in different scenario, not only in the restricted to human, but also other animals too. So as described that probiotics have some beneficial properties, so definitely all microorganisms cannot be considered, cannot be used as probiotics. There must be some selection criteria, which include, for example, the safety criteria, the functional criteria, physiological and technological criteria if you are interested to use probiotic microorganism and different biotechnological processes. So I'm not going to detail because these are very simple. I hope everyone can understand these things. Following the selection of probiotic microorganisms, probiotics perform a very huge spectrum of health benefits, not only to humans, to other animals, and to biotechnological processes in food industry, for example. So some of them are, for example, synthesis of metabolites or production of metabolites and the exclusion of pathogenic microorganisms, gut distortion, for example. These are the very prominent health benefits are associated with almost all kind of probiotics, but some of them are rare. For example, the psychobiotics, which are the used by the mental diseases, and anti-gerobiotics, which are used for the lowering or to slow down the aging process. So some of them are strain dependent, almost, although probiotics stand critically and very strictly strain dependent parameter, but some of them are shared by all microorganisms. So if you're talking about these health benefits of probiotics, so definitely in human, they are also used for different spectrums. For example, they're used for the treatment of different diseases, just indicated, ensuring that probiotics have a promising health benefits in the humans. So
I am so sorry to interrupt you, Dr. Abrar. Actually, your slides are not moving. We are stuck on the first slide.
oh, sorry, I don't know. Let me. Is it moving now? I don't know what's happened. Is it moving now?
Yes. Yeah, it's
Okay.
moving now.
I just forgot. I gave us one overlook. So these are just the content which I describe to the audience, and I don't know what happened to this. So now I'm here. Probiotic is medicine. So after describing that, yes, probiotic have promising health benefits on the human and the other animals. So can we use probiotic as medicine? So the simple answer, yes, we can use probiotic as medicine, but we cannot call probiotic medicine. There's still a debate because there is no rules proposed by FDA or EFSA or WHO, for example. So we can use probiotics for medicinal purposes, but we cannot call probiotics medicine. So what are the benefits, advantages, disadvantages? We will learn now. So I just want to clarify the difference and advantages with probiotics and medicines. For example, probiotics are live microorganism, but medicine doesn't involve the microorganism. They can obtain from different substances. Probiotics are mostly obtained from the fermented substances, but traditional medicine substances have a very huge sources. Probiotics are mostly used for the holistic health benefits. They cannot target a specific disease particularly, but medicine are particularly used to target a specific disease. If you're talking about the safety profile, so probiotics are more safer than traditional medicine. Probiotics have lesser negative reaction, while traditional medicine have a huge or more negative reaction. So this just compare the probiotic and medicine, their pros and cons. So can we use probiotic as medicine? Against the question, and the answer is yes, we can use as a medicine, but we cannot call them medicine because probiotics are frequently are commercially available as a dietary supplement or feed supplements, and they can recommend for the treatment and prevention of different diseases. And there are no available set of recommendation and no FDA guideline for probiotics available. Even the European Food Safety Agency also refused the health claim of some commercialized probiotics because of the insufficient information which are present on the probiotic products. If you're interested to use probiotics as a medicine, so what properties should be considered? These are just a very rough properties must be present almost, although the experimental validation is utmost important. For example, the strain level identification, probably by the PCR analysis or by the genomic analysis, there must be no antibiotic resistance. The strain must be safe, particularly from the human origin. It will be more advantageous if we can use a human based probiotic strain. There must be no antibiotic resistance, no virulence factor, no hemolysis activity, for example. They must have adherence, accuracy, they have viability in the once you've taken them into the host body. They also have some tolerance potential, which is most important. They can survive in the harsh condition of the gut. There must be no side effect, and they have genomic data because genomic validation, because genomic analysis is currently considered one of the most important validation study for probiotic strain, and also consumer satisfaction. Either consumer are feel safe while using probiotic or not. So if we can take a probiotic as medicine, so according to European Union regulation, there must be some information or labeling must be present in the probiotic product which are commercially available. For example, the expiry date of the which ensure the guarantee that the strain must remain viable while till the expiry date. There must be a direction of use. What are the use the route of use, the strain number, the number of CFU per gram or per ml, and other information the consumer, the targeted audience or targeted consumer or the immunification information should be present on any probiotic product which are commercially available. And we are pretty sure most of the commercially available probiotics don't contain these recommendations, so they cannot be used for the health claims or health benefits. So if we are interested to use probiotics for the medicinal purposes or for the prevention or treatment of diseases, so what are the mechanisms they are following? So general mechanism is clear, but the very we can say a very holistic mechanism they are following, or all microorganisms are following, is based on their gut restoration potential, the production of different metabolites. They can kill pathogen either by competing their binding site, or they can take the nutrients from the pathogen microorganism, or they can enhance the host immunity. So by this way, the host can fight with the pathogenic microorganism, and in this way, they can eradicate the pathogens and thus help in the human or the host health status. These are the spectrum of different diseases which are targeted currently with probiotics microorganism or probiotics. For example, the gastrointestinal diseases, the respiratory disorder, mental disorder, metabolic, allergies, infections, and all other diseases. You just count down the name of the disease, and you will find the probiotics in either are in use or in experimental condition. So if you are interested to use probiotics, there must be some guidelines should be followed. For example, the strain level identification is a crucial important. There must be complete information, for example, about the amounts, dosage, viability, storage condition, etc. There must be information about the developer and manufacturer, and there must be clinical validated study or clinical validation must be available. One thing more, which is most important in this guideline, there's the probiotic food interaction should be studied and clearly indicated, and also the strain level information should be documented. While taking probiotics, some precaution must be taken because we already stated that probiotics are strain-dependent phenomena, and we cannot say that a same strain must be safe for me and for all of us because. And the second slide I just introduced that 1/3 of the microbiota of all of us are roughly same, but 2/3 of our microbiota is not similar, indicating that we are not sure that probiotics can perform the same function of every individual or not. So some precautions should be taken. For example, a pregnant women have a chance to have allergic reaction if they can take probiotics. If some individual are in the already in chemotherapy or some treatment options, so they should avoid or should properly consult with the physician should they take probiotics or not. And they also age dependent or should same probiotics should be taken by infants or the children or the aged people. So these are some guidelines you should be followed by taking probiotic as medicine. What are the route of administration for probiotics therapy or probiotic medicine? So they can be taken by oral, vaginal, rectal, spraying in nasal, and different routes are available. There are different forms, for example, powder, capsule, granule, in spray form. The probiotics are available in the commercial form. What are the advantages of probiotic therapy? Less cost, the multiple route of administration, greater potential of adherence, less antibiotic resistance, and more safer and natural sources. So these are some of the advantages of probiotic therapy. But to confirm a strain's safety or to confirm a strain medicinal property is a tedious, time-consuming, and not an easy task, but rather a very tedious and time-consuming process. So these are some limitations also associated with these probiotics medicine. This slide just indicate the different forms in which probiotics are available in the commercial form. So following that, yes, probiotics are used currently for the different diseases, particularly in the human. So this table just indicate the different probiotic strain in the different diseases which are treated with probiotics. For example, they are help in the gastrointestinal disorder, the immune disorder, metabolic disorder, cancer, etc. This is just our published material, so don't worry, you can find all this information from our websites. So this is the different diseases which are treated with the help of probiotics microorganism. As the field of probiotic medicine is considered a very new and emerging area of research, if we're talking about the literature survey, so according to literature survey from 2000 to 2020, in 2 decades, almost 300 meta-analysis of a form which elucidate the potential spectrums or potential health benefits of probiotics. And it's very promising result that 79% showed a positive health beneficial aspects, and only 21% were found ineffectiveness or they are found negative for their health benefits. So indicating that this field is of great importance and have a very huge importance in the medicinal aspects. These are some list of probiotics which are commercially available in different dosage form. For example, Lactobacillus acidophilus are commonly used by bacteria, followed by enterococcal species are commonly available in the markets, and different probiotics in different medicine substances and used by the different treatment options. So this is just take a snapshot of the drug.com website, so you can find there more information on this website also. And this is a different medicine products which contain probiotic strain or different combination of strains. So these are the different products which are commercially available in the market, and we are also already using in different applications, particularly for the health benefits. So in summary, this slide just summarizes the pictorial summary of my presentation. The probiotic as medicine a dashboard which indicated that what are the microorganisms which can be used for the probiotic as medicine. So the strain of different species which I already described here in the this slide. And so bifidobacteria, Enterococcus, Staphylococcus, etc. The species of these genera are used for their medicinal purposes. The properties required, again, the strain level identification, safe origin, viability, and tolerance. What are the diseases treated? GIT disease, allergies, metabolism diseases are treated with these probiotic medicines. Mechanism is already described that is still unclear, but the immunity regulation or the pathogen killing or the production of different substances like antibodies and interleukin can may be the possible route of its mechanism to perform their action. Advantages include the natural treatment, safe origin, common commensal, and have vast treatment potential. While why they have some advantages, why we are not taking them as a medicine. So there are some challenges associated. For example, the safety concern may be a probiotic strain safe for me, but not for all of you, or vice versa. So they can also have some gas production, or sometimes they can also produce can kill the common commensal of the gut microbiota. So there are some challenges associated with these probiotic as medicine. What are the proposed guidelines? They must be from human origin. They have proved claims, suitable forms, and recommended dosage, clinical validation, etc. must be performed for a stand to be used for the medicinal purposes. If we are interested to take a probiotics product for medicinal purposes, so what should we, as a consumer, can identify in the product? So complete identification should be present. The recent name, the target consumer, the product potency, etc. should be present on any probiotic product. How we can overcome these limitations. So the future perspective include the accurate identification with advanced technologies, like as I told, the probiogenomic analysis or the genomic study, or the, for example, CRISPR-Cas system or the probiotic antibiotic therapy should be used to clearly indicate whether probiotic is used as a medicine or not. But these all things should be under control authority, and there should be some regulatory bodies or authorities which control these. For example, some international organization like EFSA or ISAPP or WHO, FAO, etc. are some propose some guideline for this, but there must be some country level organization should be present if as a consumer we are taking probiotic as medicine. So with this, I just conclude my presentation. These are some references from which mostly these are published material, so you can find all of these, in particularly my ResearchGate idea Google Scholar profile. Thank you. If you have any questions, so most welcome to you. Someone asked a question, can you brief us on how probiotics are they prepared and the application of taking immunocompromised individual? As I already described that there's some limitation. One of them is the immunocompromised individual. If we can, if a person which are immunocompromised status, so they are not recommended to use probiotics. Maybe or the person which have getty leakage gut or leakage intestine, so maybe they can cause some consequences. So most probably they are not recommended for this immunocompromised individual. Yes, they are used for the cancer treatment option. As I already described that, just count down the number of disease and you will find a probiotic behind them. But some of them are available in the market and some of them are not available because they're very tedious process, not an easy, as I described that, because how we can differentiate if you can, for example, take a probiotic strain to an individual and then how we can analyze the data, because the same probiotic or the strain should be present in their gut also, and we can introduce the same strain from the outside. So what is the differentiation method and how we can differentiate them? It's tedious and there is some limitation available, so we can say. Let me, if I have found more questions. So if you have any questions, you can also contact with me. My email address, or you can also message me on my ResearchGate or any of my available profile. So I think there is no question. So I will turn off my camera and stop sharing, so the host will move to the next speaker. Thank you very much. Again, if you have any questions, so feel free to ask me anytime.
Yes, you are clearly audible and your slides are visible now.
Okay, so welcome everyone. Thank you for giving me the opportunity to be a part of this event today to present my topic, Harnessing Health, a deep dive into antioxidants and nutritional profile of the Moroccan Ulvalactuca from the Mahdia Coast. This presentation aims to highlight the incredible health benefits of Ulvalactuca, exploring its nutritional composition and the role of the antioxidants. So we look forward to share valuable insights with you. To guide us through this exploration, we have structured this presentation into sections. We will begin with the introduction and objective, followed by a detailed overview of the material and the methods used in our study. Next, we will discuss results obtained. And finally, we will conclude with a summary of our finding and their significance. So based on their chlorophyll and auxiliary pigments, microalgae, often known as seaweeds, are divided into three major groups: red, brown, and green alga. These organisms are offered for sale as raw or processed goods and constitute an industry worth over $6 billion annually, according to FAO 2018. Abundance in protein, peptides, amino acids, minerals, and polysaccharides. This marine organism offer a diversity of essence. They are widely recognized for their role as a valuable source of essential nutrients and bioactive compounds beneficial for human health, antiviral, anti-cancer, anti-diabetic, antibacterial characteristics are among the functions of these bioactive nutrients. So the edible seaweed genus Ulva is an essential supper in many countries in Southeast Asia. As far as we are aware, Ulva lactuca have not been studied in the Kenitra region. The purpose of this study was to analyze Ulva lactuca approximate composition, nutritional profile, functional properties, and antioxidant properties for possible application in the food, cosmetic, dietary supplements, and healthcare industries. Now let's delve into material and method section. We will outline the key components and processes that will utilized in our study. So the first step is sample collection and preparation, where fresh seaweed was gathered by hand from the Mahdia coast, cleaned thoroughly in seawater to remove external matter such as sand and other contaminants, then placed in polyethylene plastic bags. In the laboratory, the samples were washed again 3 times with tap water and 1 more time with distilled water. Samples were dried at 40 degrees C and then kept in dark bottles at 4 degrees C after being mechanically blended into a fine powder for further analysis and application. The first one is the proximate analysis, which include several key components. First, we will measure the moisture content, which is crucial for understanding the water retention properties of the seaweed. Next, we will assess the ash, the crude protein, the carbohydrate content, the dietary fiber dosage, and the energetic value. Also, we will estimate the lipid composition by the GCMS and the mineral content by using the ICP. Finally, we will determine the antioxidant activity by using the pH and PREP assays. For the water holding capacity and oil holding capacity were measured to test the physicochemical characteristic of the powder. So the water holding capacity was only to determine the water absorption capacity, and for the oil absorption capability was calculated but using Stokesky approach. For the antioxidant activities, we employed 3 solvents of extraction: methanol, chloroform, and hexane. The pH was adjusted to assess the seaweed extract's capacity to scavenge free radicals. So the percentage of scavenging was calculated by the formula below, whereas the ABS sample is the sample and the pH solution. The ABS control, it is the absorption of the control without the sample. So the inhibition or the inhibitory concentration at which the pH radicals were 50% scavenge was measured as the IC50 value. So the extract reducing power was ascertained using the OISO method by using the potassium ferricyanide, trichloroacetic acid, and ferric chloride. Next, we will move on to the results and discussion, where we will present the key finding of our study and the application. In the first part, we will share the results obtained from our analysis. So by emphasizing moisture and ash, the alga biochemical composition was ascertained, revealing the amount of protein, fat, carbohydrates, dietary fiber, and energetic value. Ulva lactuca had a reasonably typical moisture content of 23%, and the calculated crude ash percentage for 5 grams was 15%. Protein, lipids, and carbs are among the 3 major biochemical components of seaweed. In our investigation, proteins were generally less common than carbohydrates, which represent an amount of 61%. On average, total lipids were found to be low. It represents 1.3%, where palmitic acid was dominated the saturated fatty acid composition, and the most prevalent monounsaturated fatty acid was oleic acid. In addition, polyunsaturated fatty acid were 5.5%, which consisted of a potent fatty acid in human nutrition, especially the DHA and the EPA. So seaweeds are well known for being high in minerals. In general, mineral components are necessary for both general physical and the mental health, since they are elements of blood, muscle, bone, teeth, soft tissues, hemoglobin, and nerve cells. The dominant elements determined in Ulva lactuca were potassium, sodium, and calcium. Also, magnesium was abundant. The iron levels in seaweeds were higher than in many commonly consumed sources, such as meats, because of their metabolic ability to absorb elements directly from the salt water. So for the water holding capacity measurement can be used to identify the hydration characteristic. The variation in Ulva lactuca powder's water holding capacity between 25 degrees C and 37 degrees C are shown in the figure below. So we can observe that the temperature-dependent changes in the water holding capacity. The figure displays an increase in the Ulva lactuca alga powder for the oil holding capacity at two different temperatures, as mentioned before. We discovered that when the temperature increased, the green seaweed powder oil reduced slightly. For the antioxidant activity, both in the sanders and the extracts, the activity depended on concentration. The three solvents that were utilized show difference in their antioxidant capability. So the extract has the highest capacity to absorb the PPH radicals when its IC50 value is lowest. The methanol extract was found to be notably strong and comparable to that of ascorbic acid as the positive control. All of the extracts employed in this investigation showed impressive antioxidant activity, as seen by the FRAPE assay results, which showed no negative values. The methanol extract in this study showed good first reducing activity. However, to compare the reduction activity, ascorbic acid was utilized as a reference antioxidant. So according to its physicochemical characteristics and proximate composition, Ulva lactuca has a significant amount of ash, protein, carbohydrates, fatty acids, and minerals. The seaweed that was studied is said to have a lot of potential as food supplements and might be a source of components with a significant level of nutritional value for the food sector. Ulva lactuca can value by marketing them as value added because it has been discovered to be a good source of critical nutrients. On the other hand, levels of polyunsaturated fatty acids and minerals, including magnesium and iron. For every extract, various scavenging activity levels were targeted in tests such as the PPH and FRAP assays. It was also noted that Ulva extracts potential antioxidants, which has always depended on concentration, was caused by their electron donating and free radical scavenging qualities. And thank you all for your time and attention.
We will let you what is CUX1 cut transcription factor and then glyopodia. And CUX1 basically a homeodomain transcription factor. It involves in self-head determination and dendritic arborization, and it is also acts as an inhibitor of RNA polymerase second, and it has a 4 DNA binding domain, 3 cut repeat domain, and one homeodomain, that is Hox domain. And these domains are conserved in all metazoans. For example, Drosophila. In Drosophila, it is called as a cut. In Drosophila, it has a 3 cut repeat domain and 1 homeobox domain. So so here I will show that we use Drosophila larval nervous system as a model system to study the role of glial cells. So our lab basically worked on the CNS development. So glial cells is the major component of the CNS, which provide a trophic support and other functions to the neurons and involved in the CNS development. In Drosophila, there are various types of glial cells, and these glial cells seem structurally and functionally similar to mammalian counterparts. So here you can see that this is a Drosophila CNS, and this is a transverse section of Drosophila. This is ventral nerve cord, and these are optic lobes. So here transverse section of the ventral nerve cord shows that this is a gray region. This gray region is the periphery. Here you say this gray region. This is a periphery, that is cortex, and this middle center region is neuropile region. So these glial cells are spatially localized. These two outer glia involved in blood-brain barrier formation, that is perineural and subperineural glia, and this glia, this is enseething glia. This enseething glia enwraps axon bundles, and this blue color, this is astrocyte glia. It involved in synaptic formations, and this green color cortex glia. Cortex glia basically enwraps neural stem cells and their progeny to provide support and form a neural stem cell niche for proper development. So here you can see that this structure shows that cortex glia. These green color are cortex glia, and this yellow color structure is nuclei of the cortex glia. So you can see that this single cortex glia enwrap multiple neuronal cells. So this large structure is neural stem cells and their progeny, that is neurons. So cortex glia extend a fine projections around neural stem cells and neuronal cell body. So these are neuronal cell body and neural stem cell body, basically marked with DAPI, which is a nuclear marker. So these fine projection, that is called gliopodia. So gliopodia looks like a trophosphengium like honeycomb structure. So based on that, it communicates and interacts with neurons. So our lab worked on this cortex glia and reported that when 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 cut, that is transcription factor CUX1 misregulate in the cortex glia, then the morphology of the cortex glia disrupted. So this is a cortex glia specific GAL4 driver line used for marking the cells, and here this is the experimental condition where we used RNAi, that is RNA interference, to knock down cut. So when knockdown cut in the cortex glia, you can clearly see that the morphology here, these projections clearly present, but in case of knockdown, morphology, that is gliopodia, these fine structures disrupted. Also when we overexpressed cut using UAS cut lines, so you can see that in comparison to control, here these fine projections are present and these thick projections are present but short in comparison to control. In comparison to control, here you see that in experimental condition, that is overexpression of cut, these projections extended in expense of fine projections. So it indicates that this transcription factor, that is cut, involved in glial cell morphogenesis, and it is already reported by our lab, that is cut involved in neural stem cell niche maintenance and importance for cortex glia morphology. So but still not clear how does cortex glia maintain their morphology and the gliopodia development, what pathway involved in this. So some literatures reported that is outer radial glia, which is neural precursor cells in mammals, their morphology regulated by PI3K AKT mTOR pathway. This PI3K AKT mTOR pathway regulates cytoskeleton regulators, that is small GTPages, RES GTPages, that is RAC1, CDC, and one other member, that is RO. And we analyzed using bioinformatics protein-protein interactions and found that CUX1, that is upstream of the PI3K. So we hypothesized that if CUX1 is upstream of the PI3K, and here PI3K regulates glial cell morphology. So if we misregulate these molecules, then it also regulates cortex glia. So addressing these questions, we used glia-specific genetic driver lines, that is GAL4 and QAF binary system, to misregulate uh, gene of interest and manipulate gene of interest to study the specific functions. If that proteins is not present, then what happens? And we used immunostaining and molecular assay to analyze the status of protein and expression of protein level, and super resolution microscopy to analyze image, and for morphological and behavioral analysis we used Fiji image software. So we are curious about how does PI3K overexpression and AKT overexpression affect cortex gliopodia growth when cut is knocked down. So here this is a control image, and you can see that these gliopodia, that is extensions of these cells, clearly visible. And but when we knock down cut, then these cortex glia fine projections, that is gliopodia, not developed, and that is completely lost. So when first we overexpress PI3K in cortex glia, you can see that these projections diffused and thick in nature. So it indicates that PI3K overexpression enhances or induces cortex gliopodia development. And when we used PI3K dominant negative lines, it basically completely block expression of PI3K, and it indicates that when we block PI3K overexpression using USGAL4 system, here the gliopodia, that is extensions in comparison to control, very less. And when we simultaneously overexpress PI3K in background of cut knockdown, here you can see that clearly cut knockdown phenotype epistasized over PI3K. And similarly, when we overexpressed UAS AKT, here you can see that this is the chamber, that is cortex glia chamber, where neural stem cells and their progeny present. In comparison to control, this chamber is wide and increased in membrane projection, that is gliopodia, also thick in nature. And when we knock down AKT, here chamber become smaller in size, and but there is no effect in gliopodia development. But when we overexpress AKT in background of cut, here is similar to PI3K. Here you can see that cut phenotype epistasized over the AKT phenotype. We also check mTOR RNA knockdown. When we knock down mTOR, here morphology of the cortex glia disrupted, but the gliopodia development is not as such completely disrupted in comparison to controls. It indicates that cut knockdown. It indicates that knockdown with PI3K and AKT overexpression. These does not affect cortex glia morphology. As previously, I showed that PI3K AKT regulates outer radial glia morphology with cytoskeleton dynamics regulator, that is RAC, CDC42, and RO. Here these molecules regulates actin turnover. So we are interested to check whether these molecules involved in cortex gliopodia development and cut regulates via this pathway. So, so first we check Rho expression using Rho1 antibody. So here you can see that this is a control image and these projections. So here co-localized merged image. You clearly see that there are projections of the cortex glia. These are co-localized to each other with the glial cell membrane and gliopodia of the cortex glia. But similarly, when cut overexpression and cut knockdown cases, these staining in comparison to control very low. But when we statistically analyze, it indicates that cut overexpression and cut RNAi, it is significant, but we can't say here means cut row 1 level is decreased in cortex glia due to cut knockdown and cut over expression, because here this is neural stem cells and other cells present, but here also and staining is low. But we check using row RNAi lines, what is the status of row. So this is a control image. You see membrane projections and gliopodia extensions. We knock down row using row RNAi lines. So there is no such changes in comparison to control, but number of nuclei, these yellow color structure, this red is repo antibody which marks spine glia, that is a transcription factor. So these yellow is a cortex glia nuclei. So here cortex glia nuclei number is less, but the gliopodia development is very cortex glia gliopodia is not as such disrupted in case of row knockdown. When we check WFYS RNAi, that is a downstream inducer of CDC42, because I have a no line of CDC42, so I used downstream regulator of CDC42, that is WEF RNAi. In case of WEF knockdown, there is no loss of membrane growth in comparison to control. It indicates that there is no role of WES, that is CDC42 knockdown pathway, and we simultaneously check downstream target of WES, that is INA. So in case of INA knockdown, here membrane projections, that is a large projection not disrupted as such, but fine projections, you see some fine projections are increased. It might indicate that it play a role in cortex glia fine projections development. So we check another pathway, that is EGFR pathway, epidermal growth factor receptor pathway. So we are interested, does EGFR interacts with cut and regulates cortex glia morphology or not. So here you see that this is first panel, that is control image, and when we overexpress using UAS EGFR line, the membrane growth is extensively increased, and the number of nuclei also increased in case of EGFR overexpression. When we knock down EGFR using RNAi lines, in comparison to control, here no such significant changes in cortex glia nuclei as well as membrane growth. Here you see that is cut overexpression phenotype, which is nuclear size increase in comparison to control, and membrane growth is very less, that is I previously showed you. Here when we simultaneous co-activate UAS cut and EGFR using cut overexpression and EGFR overexpression line, you see that in comparison to US cut conditions, here the nuclear size dramatically increased, but the membrane projections not such clear, because looks like GFP, which is marker we used US EGFP lines, so that is diffuse, so not clear as such. But when we knock down EGFR in background of cut over expression, so here nuclear size in comparison to control as well as US cut, the nuclear size increased, and membrane growth in comparison to US cut and control not such increased. It indicates that cut might be downstream of the EGFR, and cut might be downstream of the EGFR, and we observed that a special phenotype in case of cut overexpression and US EGFR. In cut overexpression, you see that these are optic lobes. This is a first panel control image. So here the ventral nerve cord elongated in comparison to control, and when co-activate cut as well as EGFR, you see ventral nerve cord dramatically increases, as well as these cortex glia nuclei looks like accumulated in a specific place and looks like a tumor-like phenotypes in multiple places. So it is already reported that CUX1 promotes glioma in humans. So this clearly indicates that cut with UAS EGFR promotes tumor in Drosophila CNS. So it is a good. It will become a good model for tumor study. So in conclusion, transcription factor cut regulates cortex glia morphology, and PI3K AKT pathway also regulates cortex glia morphology and gliopodia development. But cut regulates independently of PI3K mTOR pathway, and enhances cut interactions enhances nuclear size in in with EGFR overexpression, and cut phenotypes dominate over EGFR phenotypes, and co-expression of cut and EGFR promotes tumor-like phenotype, that is promotes tumor progression Drosophila.
Should we move on to the next presenter, Dr. Mohammad Sajjad Sarwar?
Yes, ma'am.
Okay, Dr. Sajjad, are you here with us?
Yes, I am here.
Oh, thank you so much, Dr. Sajjad. You are joining us from University of Okara, Pakistan. The virtual stage is all yours, sir.
My slides are visible.
Yes, sir, your slides are visible.
Yes, we see your slide.
Okay, this is Dr. Mohammad Sajjad Sarwar, Assistant Professor in Department of Zoology, University of Okara, Pakistan. Thank you very much for providing me this opportunity. The study involves the establishment of Wolbachia infection in the Aedes aegypti using the microinjection technique, and the evaluation of the general fitness of the resultant mosquito population under semi-field conditions. Okay, I'm on. Okay, World Health Organization enlisted the top 10 world health problems, and unfortunately, dengue is one of them. More than 125 countries, they have been affected by the dengue, and more surprisingly, about more than 10 epidemics and the outbreaks have been occurred in Pakistan. So far, no registered vaccine and the antiviral drug is available in the market. So the control of this dengue disease is solely depends upon the dengue vector, which is Aedes aegypti. So Aedes aegypti is prevalent in the abundantly present in the Pakistan, and it is the primary vector of this dengue virus. So all the conventional techniques which we use, they are failed to control the Aedes aegypti population. So there is a dire need to control the mosquito population using new approaches. So dengue-based strategy is one of them. Wolbachia-based strategy is one of them. Wolbachia is a gram-negative bacteria which is commonly found in the insect species, but the Aedes aegypti is free of it naturally. So Wolbachia induces various reproductive modifications in the host. For example, the cytoplasmic incompatibility, feminization, male killing, and the parthenogenesis. So what is CI? Actually, cytoplasmic incompatibility occurs when a Wolbachia infected male mosquito, it mates with the natural population female mosquito. So the resultant eggs, they will not be able to hatch. So by using this technique, we can control the mosquito population. Besides this, Wolbachia has also potential to induce other modifications. For example, it can shorten the lifespan of the host, and it also interfere with the parasite or dengue virus replication in the mosquito species. So using the, if we focus on these characteristics, so Wolbachia is ideal for the strategy to control the vector population. So what we did in this study, actually, if we succeeded to transfect the population of the Aedes aegypti by using the microinjection technique, then we can have the Wolbachia infection in the natural and the population of the Aedes aegypti, because this bacteria has the potential to transmit vertically in the next generations. So if we infect this bacteria to some of the eggs, then we can have a whole population of the Wolbachia infected mosquitoes. So the current study focuses on the transfection of Wolbachia from the Aedes albopictus mosquito. This mosquito is also commonly found in the Pakistan. So we obtained this Wolbachia from the Aedes albopictus as a donor mosquito, and we transfected this to the Aedes aegypti in this study. And after that, there are so many strains of this Wolbachia bacteria. We selected the WALBB strain, and we succeeded to transfect the population of the mosquitoes Aedes aegypti, and then we evaluate the impact on the general fitness of the resultant population under the semi-field conditions in Pakistan. We evaluated its impact on the egg laying rate, egg hatch rate, larval development, CI induction, because different strains have different potential to induce the CI, and obviously male competitiveness of the resultant mosquito species and the lifespan of this population. If we talk about the methodology, we collected the Aedes albopictus and the Aedes aegypti mosquitoes from the Lahore, Pakistan, and we reared them. And using the PCR technique, we detected the presence of Wolbachia in the Aedes albopictus mosquitoes by targeting the WSP gene of the Wolbachia. So then we transfect this Aedes aegypti population by using the microinjection technique, and then we get the P1F1 generation, which was infected with the Wolbachia, and up till the 5 generations, we also detected this infection. So this infection was successfully established. After, for the experimental purposes, we also generated the aposymbiotic line by treating the Wolbachia-infected mosquitoes with the tetracycline antibiotic, and then we obtained another line for the as a reference. So this is the semi-field condition, which this is the picture of the botanical garden of the GC University Lahore, Pakistan. So these are the parameters which we evaluated as a general fitness of the mosquitoes. So interestingly, the Wolbachia do not have any impact on the egg laying rate of the mosquitoes. So this is the in the green color, this is the Wolbachia free mosquitoes, natural population of Aedes aegypti. In the red coloring, this is the Wolbachia infected mosquitoes, and this is this group is the tetracycline treated, which means that the Wolbachia infection was removed from this. So on the right side, this is the fertility, means egg hatch rate of the mosquitoes. So interestingly, the Wolbachia have strong negative impact on the fertility of the mosquitoes. For example, naturally, more than 95% egg hatch rate is observed and in the Wolbachia infected population, about 80% eggs were able to produce the larval offsprings. So impact of WLBB on the wing size. Wing size is very important, because if we are interested to control the population in the field, so our mosquitoes, male mosquitoes, which must be able to compete the other male mosquitoes in the field, and they should mate, then we can have our target results. So for this purpose, the Size of the wing is a parameter by which we can evaluate the fitness of this mosquito. So the Wolbachia do not have any negative impact on the wing of the mosquito in the male mosquitoes and as well as in the female mosquitoes. About the appropriate mean size of the wing is about 3 mm. So we also evaluated the larval development. So it is very obvious that the Wolbachia infected mosquitoes, they have very fast larval development. It might be due to the presence of the Wolbachia, the metabolic rate, and the other functions are accelerated in the body of the mosquitoes. So on the right side, this is the cytoplasmic incompatibility evaluation. Uh, if the male mosquitoes, natural Wolbachia-free mosquitoes, they mate with the natural females, then we have about 90% of egg hatch rate. On the other hand, most interestingly, if we allowed the Wolbachia infected males to mate with the natural females, then eggs were harvested, but no egg was viable. All the eggs were unable to hatch to the larvae. So because different Wolbachia strains, they have different potential to induce the cytoplasmic incompatibility. So fortunately, this WLBB Wolbachia has 100 potential to induce CI in its host. So if we talk about the male mating competitiveness, also Wolbachia do not have negative impact on the male mosquitoes because they can compete in the field equally with the other male mosquitoes. So we also evaluated the lifespan in the semi-field conditions. About more than 55 days, some mosquitoes were able to survive. So the results above picture A and B, they shows that there is no any impact of Wolbachia on the lifespan if we offer the food to them. And in opposite case here in the image C and D, if we under the starvation conditions, the Wolbachia infected mosquitoes have very short lifespan, not more than 4 days. So in conclusion, the Wolbachia WLBB strain that has great potential to control the dengue vector Aedes aegypti population if we introduce male infected mosquitoes in the field, then we can control the population of the wild Aedes aegypti. So because it
Dr. Sujad, are you here with us?
Thank you.
Thank you so much for your excellent presentation, and your expertise were really very impactful. Thank you so much for joining us today, and we are looking forward in collaborating with you in future also. So any questions, please drop them down in the text. So yes, so we have a question for you.
Okay, thank you for the opportunity. Thank you.
You're most welcome. It's our pleasure. So moving on to the next presenter. So I'd like to introduce Fatima Cherboub from Ibn Zohar University, Morocco, who will be leading us through a fascinating discussion. So please start with your presentation.
Thank you. Can you see my screen?
Yes, we can
Hello?
see your screen.
Yes, thank you.
You're welcome.
Good evening, honorable discussant, honorable conference committee members, and all the participants. My name is Fatima Sharboub. I'm from Ibn Zor University, Morocco. Today, on behalf of all the authors to present our research proposal titled Evaluation of the Observance Characteristics of Biomass Products from Eucalyptus waste and its application for the removal of lead and cadmium. Firstly, to introduction by to the water pollution is a major environmental problem worldwide, particularly due to presence of pesticide and heavy metals. Treating this type of pollution caused by toxic substance requires complex and specific approach. The main object of this work, therefore, was to prepare biomaterials optimized through various parameters which will be characterized and evaluated for its application in observance and elimination of organic and inorganic pollutants from contaminated waters. In this slide, I like to provide some general information about heavy metals, also known as metal class element. In chemistry, heavy metals are typically defined by physical chemical properties. In soil science, trace metals are mineral components present in very low concentration. In toxicology, they are defined as metals with cumulative properties, often having a harmful effect on living organisms. In nutrition and agronomy, trace metals are not sometimes essential elements necessary for certain organisms due to their catalytic role in metabolisms. However, the classification of heavy metals is often debated. Some toxic elements are not metals, like arsenic, while some toxic metals are not particularly heavy, like zinc. The heavy metals most even considered toxic to health and the environment are cadmium, lead, arsenic, mercury, other choose of copper, zinc, and chromium. The intensive use of heavy metals is in human activities and industrial process has a harmful impact in human health and the environment. They can cause toxic disease, digestive and respiratory disorders, as will contributing to soil, air, and water pollution. Heavy metals contamination of waters can be found in different types of waters: groundwaters, reservoirs, rainwaters, and sea waters. For materials and methods, the technique used in this work is based on the absorption of heavy metals onto biomaterials containing natural eucalyptus. Then the metal concentration in the fluid were determined by atomic absorption spectrometry graphic functions. The heavy metal studies are lead, which exacted in oxidation state, and cadmium, which fuses naturally in two oxidized states. The sample is washed several times with distilled water to remove impurities and dust, then dried in an oven. The sample is grown to a fine powder using a mechanical mill and positron sieve. To biomaterials is then chemical activated with phosphoric acid. The mixture of biomaterials impregnated and phosphoric acid is filtered and dated at 110 degrees Celsius in on oven. The resulting particles are carbonized in a movable furnace then stored in a discard for future experiment. Surface morphology, power volume distribution, and power size structural characteristic and elemental composition by XRD of the biomaterial are analyzed, carried out on the powered sample. The site of techniques enables use as to conform to composition of biomaterials. For the results, we studied the effect of parameters that influence the adsorbance of lead and cadmium, shows as the mass effect of the adsorbent, the concentration effect, the pH effect, and the contact time effect. Since the presentation time is short, I will concentrate on the mass and concentration effect. The following diagram shows the evaluation of the adsorbance rate of the two elements as the function of the adsorbance dose. It shows that the removal rate reaches 19% after 0.8 GL of the adsorbent dose. The effect of the analyzed initial concentration of lead and cadmium is then examined, showing that the adsorbance capacity of lead and cadmium ions increases with the initial metals concentration. For conclusion, our study focused on the adsorption process using biomaterials, which showed excellent absorption capacity for lead and cadmium in contaminated water. Two, in addition, desorption experiment clearly indicated that adsorption of lead and cadmium ions followed one exchange and strong physical chemical adsorption. More importantly, the result of the potential ecological risk test confirmed to save application on the low cost biomaterials as an effective and environmentally friendly materials with immense potential for application in the remediation of lead and cadmium contaminated waters. For the planning future, finally competitive adsorption of cadmium and lead ion from binary metals mixtures by biomaterials used competitive Langmuir models. Study of adsorption process on pesticide by the same biomaterials to preparation of a new composite to be used for adsorption, evaluated to influence of certain parameters, shows as pH, metal ion concentration, contact time, and those on new composite, and adsorption efficiency for heavy metals and pesticides. This measurement of metals ion concentration will be determined by ICP and MS and LC and MS. And thank you for your attention.
Thank you so much for your time and expertise. Your session was truly engaging. We appreciate the effort you put it into making it so impactful. So I would like you all, if you have any questions, please drop them in the chat box until I move on.
Achha. Assalamu alaikum. My name is Dr. Saifnur Chaudhary. I'm from Bangladesh. I'm working on as a senior medical officer in the Department of ICU SDU in the Institute and Applied Health and Science Hospital. Today, my topic of presentation is correlation of hypomagnesemia with clinical outcomes in a critical ill patients admitted to a tertiary care ICU SDU in Bangladesh. At first, I would like to start by saying that magnesium is one of the most important electrolyte in our body, and it plays a role in several physiological activities. One is it modulates immune function, acts as a cofactor or enzymes, and plays a crucial part in controlling the cardiovascular physiology. It is seen that when magnesium level is low in our body, it affects our cardiac system, causing some arrhythmias in our heart, which we sometimes refer to it as long QT, and some abnormal arrhythmias happen, as I can say. And it is also frequently referred to as the forgotten electrolyte because its clinical implications and treatment are frequently disregarded despite being evident in disease diagnosis. The level below 1.7 milliequivalent per liter are referred to as hypomagnesemia, and it has been linked to increased mortality and longer hospitalizations in intensive care units and increase in the need for mechanical ventilation. Despite being the fourth most abundant cation in the body and the second most abundant intracellular cation of magnesium in critically ill patients, it is rarely considered. Hypomagnesemia is common in critically ill patients and strongly associated with disease severity and longer hospital stay and increased mortality. Our aim and objective of our study was to investigate hypomagnesemia in tertiary care ICU in Bangladesh and observe its relationship with disease severity and the patient outcome based on patient mortality and diagnosis. Our material and method was, it was a single center prospective observational study which was done on 81 patients admitted to ICU at Bahawalpur Memorial Hospital from July 2023 to June 2024. And it was seen in our result that a total of 81 critically ill patients, 40% were normomagnesemia. That means within the normal level, 1.7 to 2.2, and 32 patients were below the normal level, which taken the percentage of 39.50% of the total population, and 9 patients were hypermagnesemia. Our methodology study design was a prospective observational type, and our study population was the patients who was admitted in the tertiary care hospital in BBMH, Chattogram. And study site was the Department of ICU in BBMH, Bangabandhu Memorial Hospital. It was a 12-month long study from July 2023 to June 2024, and a total of hospitalized confirmed cases with various disease and one system organ failure was taken in our study. Inclusion criteria was all the patients ranging from 18 years and above were admitted to the department of ICU with at least one organ failure. One organ failure means a patient admitted with respiratory failure, liver failure, or more than one organ failure, that is systemic multiple organ failure such as septic shock and cardiorespiratory failure such as that. And patients.
Sir Saif, I am so sorry to interrupt you in between. Can you proceed to the next slides? Your slides are stuck at one place. That's just the title slide we can see.
Okay, I'll just, okay. Can you see now the data collection procedure?
Not yet.
Can you see now?
Yes, now we can see it.
Okay. So our data collection procedure was on the first day of patient's ICU admission. 1 milliliter of venous blood was drawn and used away for serum separation with the patient's relative giving informed consent. After being collected in a green top vacuumator, the blood was centrifuged at 3000 rapid movement per minute for 30 minutes, and 20 microliter of material was mixed with 2 milliliter of mono reagent. After shaking with a vortex, the content were intubated for 5 minutes at 25 degrees centigrade. On a spectroscope in 1 ml cuvette, the colored complex produced by magnesium and the mono reagent was measured at 545 nanometer against the reagent blank, and milligram per deciliter was used to express the result. The magnesium concentrate was measured in the normal range of 1.7 to 2.55, and the subjects were compared for a number of parameters after being split into groups that were normal magnesemia, hypomagnesemia, and hypermagnesemia. A serum level below 1.7 was considered hypomagnesemia, whereas above 2.56 was considered hypermagnesemia. In our result in this study, as you can see from the pie chart, that among the total of 81 patient admitted in Department of ICU, 40% was within the normal level, and 32 or 40% was below the normal magnesium level, and 9 patient was having high magnesium level. This is the demographic distribution, where you can see among the male and female, the magnesium level was more prominent. Low magnesium was more prominent in female, and in male, the normal level of magnesium was more observed. In the result, in our study, we used some scores for assessment of patients' outcome and well-being. One was high Apache score. Apache score means acute physiological assessment and chronic health, and SOFA means system organ function assessment and Charlson comorbidity index. Apache 2 was found in hypomagnesemia 16.96 was the mean value, and it was carrying the mortality percentage of 40%. And in SOFA also it was in 5.59, which was also between mortality of 20 to 40%. And cardiac Charlson comorbidity index was 4.75. The mean hospital stay was seen in hypomagnesemia was more than patient who had normal magnesium level, and it was in a mean value of 5.53, and whereas hypermagnesemia it was 5.7. The comorbidity of patient among patient in ICU, as you can see, patient with different risk factors. As you can see here, that diabetic and hypertensive patient had more low magnesium level as compared to other comorbidity people, whereas patient who had CKD, chronic kidney disease with end-stage renal failure, had high magnesium level. This is the pattern we have seen in patient who had mortality or patient expired in our study. That most of the patient who had multi-organ failure maintained the majority of the expired hypomagnesemia, and number of patient expired was 4 among them. And we have also found some abnormalities in our study. Was sometimes in patients with chronic kidney disease, they have also been where it is more common to be hypermagnesemia due to impaired absorption and delay excretion from the body, and there's a buildup of positive electrolytes such as potassium and other electrolytes. It was found that magnesium was sometimes found low in CKD patients, and among the 32 patients, patient with hypomagnesemia, 11 was expired. So in our conclusion, we have seen that the effect of hypomagnesemia in critically ill patient was highlighted, and it was well correlated with more adverse scenarios, showing a positive relationship with mortality and longer hospital stays. A positive relation with disease severity was also seen with significant Apache, SOFA, and Charlson comorbidity index in hypomagnesemia patient as compared to patient with normal magnesium level. So I would like to thank everyone for listening to my presentation. If there is any query, you can ask. Thank you.
Thank you so much, Sir Saif, for leading us through your presentation. That was truly enlightening. So, any questions? All right, let me move on to the next presentation. That is going to be from Abida Barai, and she had been really patient with us throughout this session, and she had been waiting for her turn, and we are also waiting to hear from you. So, Sir Seth, would you please like to stop sharing your screen so that Abida can share her screen? Thank you so much. So, Abida, if you are ready.
Yes, hello everybody. Before I start my presentation, I would like to thank the organizers for making this event possible, and it's really a privilege to be a part of it. So, can you see my screen?
Yes, we can see your screen, and you are clearly audible too.
So my name is Bara Abeida. I'm a PhD student in biology at the University Hassan II of Casablanca, Morocco. So I'm honored to share with you a part of my work which focuses on promoting biocides from natural resources to minimize environmental pollution, the case of lemon verbena. So here is the presentation outline, background of the study, introduction, materials and methods, results, conclusion, and perspective. Let's start with the background of the study. So we live in a time when the impact of synthetic chemicals on our environment and health is becoming increasingly concerning. The pesticides widely used to protect crops and stored products are among the main pollutants. They contribute to the contamination of soil, water, and air, while also leading to resistance in harmful organisms. So in this context, the use of natural biocides remains a necessity. Biocides derived from natural resources offer a viable and sustainable alternative. Unlike synthetic chemicals, they are often biodegradable and have a lesser impact on ecosystems. Furthermore, they are generally less toxic to humans and non-target animals. Promoting natural biocides is not limited to the ecological benefits, it's also an economic and opportunities, like encouraging the local production of biocidal plants can create jobs and revitalize rural economies. Also, farmers can reduce their costs for plants protection products while improving the quality of their products. Introduction. Kelpie weevils are harmful insects of the brushwood family that attack legume crops both in the fields and during storage. These insects cause significant qualitative and quantitative losses, which can reach up to 100% of the stored grains. To counter these attacks, insecticides are used, although this solution has several drawbacks. So the disadvantages of insecticides are development of resistance, impacts on biodiversity, contamination of water and soil, and risks to human health. So, cowpea weevils, also known as the four-spotted weevil or Calosobruch maculatus, is a species of beetles belonging to the family Chrysomelidae and the subfamily Bruchinae. This species has spread worldwide and now infects legumes. The complete life cycle from egg to adult can be as short as 3 to 4 weeks under optimal conditions. These allow cowpea weevil to produce several generations per year, typically 6 to 7 in tropical climates. Adults move quickly and can infest file seeds and then can also continuously reproduce in stored dried peas. Let's talk about the lemon verbena. Also known by the scientific name Alosia trifila, is a plant of the Verbanaceae family. It is native to Chile.
Is our presentation visible? Okay, so dear conference speakers and presenters, participants, we are very pleased to present you our scientific work entitled Comparative Analysis of Flowers Sold in Azerbaijan under the name chamomile. And the presenters are, which is me, Jahan Hussein and Jale Abbasov. So this is our plan. First, we will talk generally about Matricaria ricucita, then about the purpose of our work and analysis, macroscopic, microscopic, TLC, thin layer chromatography, spectrophotometric analysis, and extraction of essential oil. So the object of our research is Matricaria chamomilla, which is the official chamomile, and it belongs to the family Asteraceae. The raw materials that are used are flowers, and composition includes such substances as flavonoids, derivatives of apigenin, which is apigenin 7 glucoside, and some essential oils. Now let's talk about the relevance of our work. So far as it's known, chamomile has been the most widely tested medicinal plant since ancient times. And there are 3 synonyms for it, which are Matricaria ricucita, Matricaria chamomilla, and Chamomilla ricucita. And there are several genera of plants that are similar in appearance to it, that are purchased in markets and or online, but they have different chemical composition. And they do not undergo proper quality control, which is why there is a risk of replacing chamomile with one of these impurities. And as a result, chamomile raw materials are replaced by other species, similar in appearance, but different, as I already said, in chemical composition. So now about the purpose of our work. It is a comparative pharmacognostic analysis of official chamomile and raw material purchased at the city market. As it can be seen in our slide, there are 2 raw materials, A and B. A is the one that was purchased from the pharmacy, an official chamomile, and B was purchased at the city market, collected in the Esmali region of Azerbaijan. It is actually also shown in like our card map. Yeah. So let's talk about analysis. First is macroscopic analysis. So we are mentioning here organoleptic analysis, which is smell. It can be seen that in raw material A, it was specific, in B, it was herbal. Then the size of the floral diameter. In A, it was from 4 to 20 millimeters. In B, from 8 to 30. And the longitudinal section of the receptacle in picture A and species A, it was hollow and conical, but in B, it was filled and spherical. Next analysis that we did was microscopic. As you can see from the pictures here, we made analysis of tubular and leaflet flowers. So picture A, you can see elongated epidermal cells of tubular flowers with slightly sinuous walls and multiple stomata. In B, cells of the epidermis of the inner side of ligulate flowers with papillary outgrowths. C is calcium oxalate crystals. D and E are glandular trichomes, and F are pollen grains. This is a bigger picture. Here we can see that there are glandular trichomes in side view, top view, and stomatal apparatus. So the specific structure of glands is a distinguished characteristic of the officinal chamomile. So as you can see in raw material A, there are cells, there are several rows. And also in raw material A, we have seen that there are more of them than in raw material B. As for the stomata, also the number of stomata was more in raw material A than in raw material B. Now, if the work will continue, Jale Abbasova. And the next stage of comparison was similar chromatography. We made extraction of two types of raw materials, which were chromatographed by similar chromatography in the butanol acetic acid water solution system in a ratio of 5, 1, and 4. The plates were visualized under 365 nanometers long wave UV light and 20% sulfuric acid heated to 100 degree for 1-2 minutes. As can be seen in the photo, the chromatographic profile of sample A and sample B differs. In sample A, we can observe a greenish spot with a ref 2.0 corresponding to apigenin 7 glucoside, which is not observed in sample B and which meets the requirements of the European Pharmacopoeia. And in addition, the quantitative determination of the sum of flavonoids in both types of raw materials was carried out using the spectrophotometric analysis. For this purpose, solutions were prepared according to standard procedure, and we used a routine solution as a witness of the standard solution. The obtained spectra allowed us to calculate the sum of flavonoids in each solution in relation to routine. So in sample A, it was 2.3%, and in sample B, it was 2.4%. The total sum of flavonoids in both types of raw materials are close to in percentage content of flavonoids. And the final stage of comparative analysis was to obtain essential oil from both types of raw materials using hydrodistillation. So to do this, 150 grams of reduced raw material was soaked in 2 liters of water and hydrodistilled. In the photographs, you can see that the essential oil from sample A has had a blue color, which confirms the presence of hamazunen in raw material, but we didn't get such a result with sample B, which was required to be proven. So
now,
Thank you so
I
much
was
for
there.
your cooperation. Okay, thank you. Okay.
Yes, ma'am. Thank you, ma'am. Ma'am, I was presenting high temperature stress. As these changes pose a significant threat to the sustainability of the approximate 2.8 billion people living in rural areas. Plants are sessile, that is, they cannot move from one place to another, which makes them incapable of escaping high temperature stress. By simply moving to a suitable stress-free environment like humans, we use ACs, fans, etc. As a result of climate change, it is pivotal for plant scientists to evaluate strategies to reduce the impact induced by high temperatures in different crops. According to a report, the map of world is shown in red, where massive decrease in crop yields are projected to occur in 2071. Parts of America, South America, West Africa, and South Europe, India, and China are being seen here affected. This credit goes to NASA, Katie Marsimam, its team, who got this picture in front of us. Now it's melatonin. Bovine pineal gland was the site of initial discovery of melatonin. Its derivative was tryptamine, which is an indolic compound generated from tryptophan. Studies have conducted to elucidate biosynthesis processes in both animals and plants. Melatonin has a natural solid antioxidant potential in stressed plants. Melatonin is an emerging plant growth regulator and is recently known to play a significant role in repairing and mitigating high temperature mediated plant variations. Melatonin regulates several morphological, physiological, and biochemical aspects under biotic and abiotic stresses. I have made a diagram here to show in crisp that here melatonin is has been implemented and given exogenously, and here we can see that endogenous melatonin is upregulated, and so many functions are going here. There is cross-talking between auxin, gibberellin, abscisic acid, cytokinin, GSSH, and ethylene, jasmonic acid, etc. Effects of plant during high temperature stress. Oxidation of proteins happens, membrane damage happens, redox imbalance and decrease in photosynthesis, increased senescence, reduced nutrient uptake, osmotic stress, denaturation of proteins, water deficiency, stunted growth, reduced crop yield, and global food insecurity at last. Colon abortion also happens there. And melatonin has a capability to mitigate all these stresses. Now, Solanum lycopersicum, that is tomato. Commonly known as tomato, belong to the Solanaceae family. It typically grows to length 90 to 180 centimeter. Tomatoes are widely utilized in culinary dishes, salads, sauces, and juices, as we all know. Their relatively short lifespan and amenability to genetic manipulation make them ideal candidates for experimental studies in plant genetics, physiology, and biotechnology. Researchers choose tomatoes for their diverse traits, facilitating investigations into disease resistance, fruit development, and environmental adaptations. Now, these are objectives in my PhD, in my research studies, to study the response of temperature sensitive and tolerant varieties of Solanum lycopersicum to different concentrations of melatonin, to study the impact of different concentration of lead in two different varieties of Solanum lycopersicum. We are also working on heavy metals, that is lead, and we are working on role, that is the study to role of melatonin high temperature on two contrasting varieties of Solanum lycopersicum. I have chosen this experiment here. A pod experiment was performed in the net house of Department of Botany, Aligarh Muslim University, to study the response of temperature sensitive and tolerant varieties of Solanum lycopersicum to different concentration of melatonin. It's worth mentioning here that one of my senior has already worked on 10 varieties of tomato in the same department under the supervision of my research supervisor, and he had given me two varieties of tomato, that is S22 and PKM. S22 is temperature sensitive variety, and PKM is temperature tolerant variety. I have chosen those two varieties for my research study, and this is the plan pattern. Temperature sensitive variety, that is S22, and temperature tolerant variety is PKM. And I have given 3 or 4 of treatments here: control, melatonin 20 micromolar, melatonin 50 micromolar, and melatonin 75 micromolar. And I have studied so many parameters. Some of them I mentioned here, that is root length, shoot length, root fresh mass, and shoot fresh mass. We can clearly see that when I have given 4 treatments showing effect of 3 different concentration of melatonin, 25 or 50 or 75 micromolars here, other than control, on shoot length, root fresh mass, root fresh mass on 2 varieties of Solanum lycopersicum. We can see here that in control, we can see that according to our studies, and we can in these results, we can say that melatonin is a plant growth regulator. We can here we can see in this slide that root length, shoot length, and root fresh mass is going to be upregulated here. Shows increase here. Shoot dry mass, root dry mass, leaf area, and spread values are here shown. I have also studied net photosynthetic rate, stomatal conductance, carbon dioxide conductance, transpiration rate, etc. And this is the statistical analysis of those very parameters which I cannot mention here. Likewise, here shoot length, shoot dry mass, root dry mass, spared values, Fe beta Fm, protein, proline, POX, SORT, CAT, H2O2, and O2. These are all POD, SORT, and CAT. These are some antioxidant enzymes which shows upregulations when melatonin is given to stress plants. It clearly shows that melatonin is an antioxidant. It acts as an antioxidant molecule. Our conclusions: significant improvement in growth, photosynthesis, biochemical attributes of tomato plants were observed against control in all the treatments of melatonin applied, as you have seen in previous slides. In foliar treatment of 3 different concentration of melatonin, that is 25 or 50 or 75 micromole, significant improvement in growth, photosynthesis, biochemical attributes of tomato plants were observed among 2 different varieties. The order of results of different parameters come as 50 micromolar being very good and shows good increase in all the parameters, which is greater than 75 micromolar, which in turn is greater than 25 micromolar, and which in turn is greater than control. As a result of the different concentration of melatonin applied through foliar mode, 50 micromolar comes best. Now, remaining objectives, I will study in my ongoing PhD different concentration of lead in 2 different varieties of Solanum lycopersicum melatonin under high temperature stress in contrasting varieties. 2 studies, the study of role of melatonin in contrasting varieties of Solanum lycopersicum under lead stress, the study of role of melatonin on 2 contrasting varieties of Solanum lycopersicum under lead and high temperature stress in common. Thank you. And I am thankful to all of you for listening me and having come listening my presentation. Thank you.
Thank you so much, Barkat. And your presentation was incredible. Your knowledge was so deep, and your passion comes up with your presentation. And I personally appreciate your presentation skills, your clarity of speech, and your knowledge of your presentation. I really appreciate you. Thank you so much for your patience, and thank you so much for joining us today. Dear audience, if you have any questions, please drop them down in the chat box until I move to the next presentation. So
Thank you, ma'am. Thank you.
it's our pleasure. It's our pleasure. So moving on to the next presenter. Uh, do we have Hakima Farahi with us from Ibn Zohar University, Morocco? Are you here with us?
Hello. Hey, hello.
Yes. If you're here with us, then please you can
Yeah.
start with your presentation.
Okay, can you hear me?
Yes.
Yeah, that's good. Okay. Good evening. My name is Muhammad Omar. I am head manager of biotech and medical science in CXI Technology Company in Cyprus. Also, the vice manager of biotech and medical science in the National College of Surgeons in USA. I am honored to be today with the National Conference of Advanced Life Science to share my insights in the antibiotic resistance in terpenes and or in the well water. So we can gather here and united our passion to the identification and antibiotic resistance in well trees to collect experts and experiences can lead to magnified discussion and innovation. Okay, and let's see, we can share our research. Our research, this is the research of incidence and identification of terpenes in Morocco. This research was cooperation with Rabat University, and here we can saw and covered the research paper. Today we can talk about water quality and health risk. We emphasize the critical role of water quality in public health in developing region. Contaminated water sources can lead to spread waterborne disease. Here we can focus on Salmonella species. As we know, Salmonella is identified as a major pathogen associated with food and foodborne illness. We here discuss the global health of Salmonella infection that can lead to severe gastrointestinal disease and complication. In our research, highlight the growing problem of antibiotic resistance among bacterial pathogens, including Salmonella, and the resistant strains complicates treatment option and poses significant challenges. Here we know antimicrobial resistance. Antimicrobial resistance poses a serious threat to human, as we know. It's a real threat to human health, making it more difficult to treat an increasing number of illness. Here we driven up the cost of care. The most popular and extensively used of antimicrobes that causes bacterial cell lysis, as we know, cell lysis of bacteria, are beta-lactam antibiotic, which inhibits the production of bacterial cell. Also, and there is a chemical structure. This antibiotic have been derived in several groups. As we know, there is a several groups of antibiotic like beta lactamase inhibitors, beta carbapenems, cephalosporin, and penicillin 19. So we conducted on Delta Plain, Morocco. This area, why we choose this area? Because have an extensive agriculture practice and underscores potential and agriculture runoff and water management to contribute the contamination of soil water. So our aim of study to assess the incidence of salmonella in well waters and the determination the antibiotic resistant profiles of the isolated strains. So let's know how to conclude by the importance of this research. We aim to contribute our efforts to improving water safety and public health. Then we have methods in this. We take in our methods. We, as we said, we work until the plain of Morocco. This region have an agricultural activities and the reliance of groundwater for irrigation and drinking purposes. Specific attention was given. We focus in two locations, Bani Musa and Bani Amir, and the collected waters from these two areas. How to collect our sample? We collect our samples from a variety of wells to ensure the representative analysis. We use a sampling protocol like collection different season to account for potential variation and decontamination level. We use sterile containers to prevent contamination. We have a detailed records for the sampling location and environmental condition to correlate with results later. Also, we use some microbiology analysis like enrichment culture. As we know, water samples inoculated with enrichment process, selective plating, like sample platted into selective media. Also, we do biochemical testing with isolation columns underwent biochemical tests to confirm the identity of Salmonella species. Look, we are antibiotic susceptibility testing. We assessed with the disk division method, which involved with selective range of relevant. We use relevant antibiotic. We also inoculating Muller-Hinton agar plates, isolated Salmonella strain. Then interpreting results based on established guidelines, CLSI standard, to determine the resistance of our research. So we use statistical software to determine the Salmonella and the pathogens, and also if any correlation between the presence of Salmonella and the environmental factor, including geographical location and seasonal variation. Okay, then we use a quality control. Okay, quality control to ensure the reliability of the results. This study we use quality control to measure regular calibration, control strains, replication tests to confirm findings. Then we find in our key findings that the study the significant presence of Salmonella in well water from both Bani Amusa and Bani Amir, indicating concerning level of contamination. We have do analysis of season fluctuations in Salmonella prevalence with higher detection rates in certain times of the year, likely agriculture practices and rainfall patterns. Also, suspension populations of isolation Salmonella strain accepted resistance to multiple antibiotics. Common resistance included ampicillin, tetracycline, and sulfamethoxazole. We identified the specific resistant patterns among the strains and also do a potential for horizontal gene transfer and the influence of antibiotic usage of agriculture that observed resistance. We uses 380 and 0.7 resistant for 6 antibiotics and also 19.5 for resistant of at least for 9 antibiotic. This leading to how to use or finding highlight significant public health concern. We, as a presence of antibiotic resistant salmonella in drinking water poses risks for human and food safety. Environmental factors, we emphasize the role of environmental factors such as agriculture runoff, waste management, that's contributing to the contamination of groundwater. So our necessary in this research to monitor the water quality and the implementations of effective water management. Also to care about policies recommendations of this study of antibiotic resistance and improving water safety, particularly in agriculture region. Then we recommend for all authors to explore the genetic mechanism of resistance and assess the impact of environmental interventions on reducing contamination levels. In this section, we show our research and how antibiotic resistance effect of public health. Thanks for your attention, and if anyone want to discuss about this research, he can contact me with WhatsApp or email. Thanks for everyone.
I would like to call upon Hakima Farahi if she is with us from Ibn Zohar University, Morocco. Okay, I suppose she is not here with us, so I will move on to the next presenter, that is Oladele Efulova Anike from Ahmadu Bello University, Nigeria. Are you here with us?
Yes.
Yes, ma.
Oh, I'm so sorry, Farahi Hakim. I'm so sorry. I thought you're not here with us. You can start presenting. Thank you.
Do you hear me?
Yes, we can hear you clearly. And your slides are also visible.
Do you see this slide?
Yes, we can see your slides.
Yes. So, honorable discussant, esteemed members of the conference committee, distinguished participants, good evening. Good evening. My name is Hagima Farahi, and I'm from University Ibn Zar University, Morocco. Today, it's my privilege to speak on behalf of all the authors to present our research proposal entitled Removal of 2,4,6,3 triazine solution by anodic oxidation using a boron diamond anode.
in this slide, I will give you introduction. So water pollution is a major problem, especially water-loaded with pesticides. This water can therefore be treated using advanced oxidation process AOP, such as electro-oxidation, photophantom, electrophantom, etc. This process destroys persistent organic or organic pollutants in carbon dioxide, CO2, and water, H2O, and inorganic salts. So we have organic pollutants or pesticides, and we have hydroxyl radical. Now this OH pound attacks pesticides with these organic pollutants and form some intermediates here, and in the end complete mineralization in water and carbon dioxide. In this slide, I will give you some generality of pesticides and the type of pesticides and why the agriculture use some pesticides in agriculture.
I am so sorry to interrupt you in between, but we cannot see your presentation. If you can please share your screen with us.
I can't hear you. What?
We cannot see your presentation. Can you please share your screen with us?
And now? You see?
No, it's not. It's not visible.
No?
Unfortunately not.
I don't understand why.
It's okay. You can continue without sharing your screen.
Yes.
Yes, it is visible now.
It's visible now?
Yes, it's visible now.
Okay. In this slide, I will to give you some generality of pesticides and why the agricultural use some pesticides, type of pesticides, etc. The pesticides can use it to repel, destroy, or combat harmful organisms or parasites affecting a crop, or increase production yields by ensuring sustainable protection of the plant. In this slide, I will to present the impact of pesticides on health, like toxicity, disease, digestive and respiratory disorders, reproductive disorders, and the effect or impact of pesticides on the environmental, like degradation of agricultural soils, atmospheric pollution, and water pollution. Remove two material and methods. Material and methods. In this slide, I present the device or step or setup used include electrochemical cell, DDB, potentiostat, and pump loading air into the solution. After degradation, I take this sample and run them through the spectrophotometer UV visible and measure the absorbance and calculate the removal percent rate. In this slide, we can see diagram of absorbance band evolution during triazine degradation as a function of electrolysis time. We can see in this slide the band of our pesticides in 0 minutes and his evolution during time. In the beginning, we have two bands sorry, his evolution with we can see in this slide the two bands of our pesticides in the first, and his formation of intermediate just after 50 minutes of degradation. Remove to degradation of pesticides. The following figure shows the evolution of removal rates of our pesticides as a function of time. It shows that the degradation rate research 99.9% after 40 minutes of electrolysis by anodic oxidation. In this slide, I present the effect of pH of the removal of pesticides. We can see that pH equal 3 is our optimal value. And in this slide, we can see effect of current on the removal of pesticides. Effect of current imposed on the removal of pesticides. In this slide, we can see the effects of current imposed to removal. So the increase in current applied or imposed accelerate the rate of degradation or removal of our pesticides. In the end, the degradation of this pesticide by anodic oxidation depends on pH of the reaction medium, concentration of the pollutant, intensity of current imposed, and some perspective or future planning, comparative study of advanced oxidation processes, AOP, optimize the experimental conditions for pesticides mineralization, detail the reaction mechanism for the degradation of this pesticide, identify the derivative or metabolites of these pesticides. And thank you for attention.
Thank you so much, Faraya Kima, for your insightful presentation. It's my humble request to all the upcoming presenters to please keep it a little short and brief, as we are running out of time and we have like a few of the presenters left who need to present. So, Faraya, will you please stop sharing your screen so that I might be able to call the next presenter? So we have Oladele Efulova Anike from Ahmadu Bello University, Nigeria. I hand over this virtual stage to you. Please start presenting.
All right, thank you, Ma. So I am Sarar Zdaq from Ibn Tufail University from Morocco. I'm delighted to welcome you all and to have the opportunity to share my research finding at this international conference. It's an honor to participate in this event, even online, and to connect with the experts from diverse backgrounds as we exchange insights and advancements in our respect fields. Thank you all for joining, and I hope this presentation will capture your interest. Today, we will be exploring nutritional composition, functional and chemical characterization of Moroccan Opuntia ficus indica cladode powder. Covering the objectives, methodology, and main findings of my study. Since ancient times, our grandparents have used medicinal plants for therapeutic purposes, and then they discovered that these therapeutic properties are due to the presence of certain secondary metabolites that are accumulated in the different parts of the plant. Opuntia ficus indica is a Mexican plant known in a large number of countries for the traditional and popular use of its various parts. The prickly pear is best known as a fruit crop and play an important role in human nutrition. It's also used as fodder for livestock. For my objective, this current research focuses mainly on studying the composition of natural origin. For this reason, we begin by identifying the chemical, functional, and nutritional composition of the plant. Then a comparison of the composition between the three study sites. For the material, plant material, it was collected from three sites. The first is the one Oriental region, and second the province of Al Husayma, and the third in the Southern region. After manual cleaning of the cladodes, they were dried and then ground to obtain a fine powder. When then determined the nutritional composition to assess pH, moisture, ash, carbohydrates, fat, and fiber content, we carefully followed the traditional method established by AUAC method. Was assessed using the Lowry method for protein quantification, and the total content was measured in fresh cladode. We quantified the mineral composition of the buffer powder using ICP spectrometry. We analyzed the surface function using FTIR spectrometry. The properties, we have determined the water holding capacity, the oil holding capacity, and solubility index. For our results, as shown in our graph, the chlorophyll content between the Imzuren and Oulad Boubkir sites was quite similar. However, the Skura sites displayed a statistically significant difference compared to the other sites with a high chlorophyll content. The chlorophyll content of cladodes is directly linked to the increase in photosynthesis
as they age and at the start of the fruiting process. For the proximate composition of Opuntia ficus indica cladode from 3 Moroccan sites, estimated in this study, varied between 99 and 92%. In fact, cladode are exceptionally rich in water. The acidic and significant pH value of cladode varied between 4.51 and 4.05. This acidity contributes to the inhibition of bacteria growth. Ash analysis revealed a very similar insignificant value between the two regions, Ouled Boubkir and Imzouren, while a significant value of 24% was recorded in the Skoura site. Regarding carbohydrate, the analysis revealed insignificant values between the 3 study sites. The fat content was observed to be low and showed insignificant difference between the 3 sites of Imzouren, Ouled Boubkir, and Skoura. For protein, our study indicated insignificant values from Ouled Boubkir and Imzouren, while the value found in the Skoura sites is statistically significant compared to the other. The crude fiber content and soluble fiber in dried cladode showed insignificant difference between the study sites. With regard to the mineral composition, the cladode from different sites, potassium appears to be the most abundant mineral in cladode, followed by magnesium, calcium, sodium, and low level of iron and zinc. A notable difference in composition was observed between the 3 sites, and the minerals are widely recognized for their essential role in human nutrition, contributing to overall physical and mental well-being. For the analysis of surface functions, the FTIR spectrum was analyzed within the wavelength range of 40,000 and 400 centimeter lines to characterize the main functional groups present in cladode powder. The result shown a series of characteristic bands corresponding to the functional groups present in sample analysis. A board peak was attributed to hydroxyl groups linked to alcohol, phenols, and carboxylic acid. Vibration of aliphatic carbon-hydrogen bonds, and presence of allicane bonds specific to Skoura and Imzouren samples. A notable bond associated with carbonyl groups, and the presence of carboxylate double bond of deprotonated carboxylate groups. And presence of polysaccharide groups. Finally, band corresponding to stretching involving halogen. This spectra reveal the presence of various functional groups, highlighting the complexity of samples composition. For the functional propriety, are primarily influenced by the fiber found in the powder. For the water holding capacity was highest at the Skoura site, measuring 174 gram of water per 100 gram of dry matter. These results show a notable difference for Skoura with a significant higher value than the other sites.
In terms of oil holding capacity and solubility index, our investigation found insignificant values across all three study sites. It's important to note that solubility index correlates with the presence of soluble molecules in powder. To conclude, a differential distribution of the chemical composition of cladodes according to region was confirmed by nutritional, mineral, and functional characterization and functional group of cladode. And as an aside, we're going to try another extraction method to apply this extract in biological activity and adding value to plants in the agri-food and medicinal sector. And thank you for your attention.
Thank you so much, Sarah Razak, for such an insightful presentation. And thank you for joining us today and waiting so patiently for your turn. Very quickly, I would like to move on to our next presenter, which is Pujita from University. You're most welcome, ma'am, and it's our pleasure. So we are going to call upon Pujita from University of Science and Technology, Bangladesh. If you are here with us, kindly start with your presentation. And I would like to request all the remaining presenters to keep their presentations short and concise because it's already too late. So Pujita, if you are here with us, you can start with your presentation.
Yes, thank you so much. Okay, can you see my slides?
Not yet. We can hear you.
Okay, it's presenting.
Yes, we can see your slides now.
Okay. Okay, is it moving? Wait a bit. Can you see it?
Yes, we can clearly see it.
Okay, so greetings to everyone. My name is Pujita Shil. I am from the University of Science and Technology, Chittagong from Bangladesh, and I will be presenting immune responses and molecular insights of dengue fever, which is a systematic review. So we know that dengue mosquitoes have become the cause for concern multiple times, hitting the headlines for various diseases. In this case, dengue is becoming challenging. Why is that? Because the virus is constantly evolving to stronger versions of genotypes and serotypes that become unrecognizable to our immune system. So more than the clinical management, our focus should be more on the molecular behaviors of the disease state. It also becomes mandatory to manage strategies for prevention, control, and protection. Generally, dengue is treatable but has the potential to develop in severity of the disease. To understand this severity, first we need to know its characteristics, which leads our understanding of molecular linking to the manifestation as we go through these 4 sections of the presentation. So, mosquitoes have a vectorial capacity to contain and transfer virus. They have been the top reason for the range of diseases from which malaria, dengue, and chikungunya are commonly known. It was present from earlier times when dengue was referred to as water poison, as open source water tends to be its breeding ground. Its fever phase had the description of breakbone fever because bone marrow is assumed to be the major target organ for the dengue virus infection. Interestingly, in the US, dengue was known as the fever in the returned traveler due to its appearance only possible in travelers who had arrived from dengue endemic regions such as Southeast Asia and the Caribbean. But at present, dengue has become endemic in more than 100 countries around the world, especially in temperate and moist zones. The severe form of the disease was first reported to be possibly in the Philippines, and around the 1960s it was recorded as Dhaka fever in my country, Bangladesh. At present, dengue is on the rise due to the unattended and untreated water sources that are in numerous locations in developing cities. Aedes mosquitoes are extremely common, becoming the principal vectors of dengue disease. Moreover, unfavorable urban infrastructures like the lack of piped water systems are a huge contributor. Increased travel, such as business trips and vocational tours, among these endemic regions is increasing the transmission of dengue even rapidly. To add to the problem, there is no specific age or gender, but groups including the working class, students, the very young, and the elderly are at high risk. Here we can see a flow diagram of the development of clinical manifestation of dengue fever. It begins when a dengue virus infected mosquito bites an individual and transfers the virus into the skin through this blood meal. The virus multiplies in the skin resident cells where the primary innate immune cells defeat, detect and activate releasing interferons and cytokines. However, during secondary heterotypic infections, when these cells fail to kill the virus completely, the virus and the infected cells enter the lymphatic and the circulatory systems through the medium. The virus succeeds to establish viremia or higher viral replication, initiating activation of immune responses specific to the dengue virus that include precise signaling pathways that raise the white blood cell counts and biomarkers of the damaged sites of the organs. In this war against the virus, the body suffers high blood pressure, rashes, respiratory trouble, and bleeding and organ impairment and dysfunction in severe forms of the disease. So how does the fever transforms to severe dengue? The answer to that could be in the molecular section of pathogenesis, for which a general idea of the anatomy of the virus becomes helpful. There are three folds to it: structural, genetic, and functional. The structural part consists of the envelope and membrane protein, viral genome, nucleocapsid, and envelope and genetic materials that code for the non-structural proteins also that have dengue relevant functional features pertaining to the replication and enzymatic processes regulated by these seven proteins, which are NS1, NS2A, NS2B, NS3, NS4A, NS4B, and NS5. In this slide, we have a general view of an immune signaling pathway that includes extracellular receptor, intracellular sensors, adapter proteins, and genetic material. The pathogen associated molecular patterns, PAMPS, and could be the whole virus or parts of the virus that present as pathogenic to our immune system. The molecules undergo modifications such as phosphorylation, dephosphorylation, ubiquitination, etc., that relay the significant particle complex into the nucleus that ultimately regulates the genetic material to produce the cytokine proteins. These are the principal cells and molecules of the host cell machineries that include T cells, B cells, dendritic cells, immature dendritic cells, monocytes, mast cells, macrophages, and Langerhans cells, each playing extremely significant roles during the infection. The major pathways are JAK-STAT, TLR, RLR, Fc, gamma receptors, and C-gasting. This is the overview of the cellular activity during the infection. At first, the immune cells activate and fight the virus systematically, starting complex series that get initiated on contact with the PAMPS. As we said earlier, the free virus and the infected cells enter the circulatory systems that eventually attack and invade the major target organs: liver, spleen, and the bone marrow. In an immune cell, for example, a dendritic cell can experience a combination of a series of the activities of complex pathways to produce specific interleukins and interferons to inhibit the virus, regulate the infected cells, and activate other cellular processes in other immune cells. Even after such complex and accurate measures of our host machineries, the dengue viral elements have the tendency to interfere by blocking the phosphorylation, dephosphorylation reactions, enzymatic processes, modifying signaling molecular structures, disguising as host materials, and promoting inhibitory reactions that ultimately block the signal and cannot destruct the virus. In other words, for the disease development, two factors belonging to virus and host are responsible. The viral factors are damage and interfere the host activities by the structural and functional elements that genetically influence and create virulence, whereas the host develops such inhibitory actions that gradually tend to destruct the host body itself, that develop into errors such as the overproduction of cytokines called cytokine storm, original antigenic sin when the initial T cells are cross-reactive to the different serotype and do not entirely neutralize the virus. These errors can lead to plasma leakage, organ failure, thrombocytopenia, that is low platelet levels, and circulatory failure. Finally, we learned that in primary infection, both innate and adaptive systems can fight and cause general symptoms of fever and memory cells against the primary virus. But it gets challenging when the virus gets past both of these systems, creating misalignments such as abnormal T cell activation, cytokine overproduction, insufficient antibody action that cause severe dengue, for which we only have symptomatic care. For these molecular interferences, we require more studies focusing on processes that target significant reactions and machinery locations for effective vaccines and medications. And with this, I end my presentation here. Thank you everyone for your kind support and patience and for organizing such an intellectual event that has allowed us to collaborate with other talented scholars from around the world. Thank you.
Thank you so much, Dr. Pujita, for joining us here today.
You're
And your
welcome.
presentation was full of awareness. I literally loved your presentation skills, and we look forward to collaborating with you in future also. Very quickly, I would like to move on to Makbul. She is from Government College University, Faisalabad. Javeria, if you are here with us, let's start with your presentation, please.
Assalamu alaikum.
Walaikum assalam, Javeria.
Can you guys see my screen?
Yes, we can see your screen.
Okay, Assalamu alaikum. Myself, Javeria Makbul. I am from Government College University, Faisalabad. I'm a PhD student in biochemistry. Today, my topic of presentation is binding affinity of beta vulgaris with HB by molecular docking and evaluation of effect of its selected phytochemicals. First of all, we will start with basics that hemoglobin is a main constituent of our blood. Tetrameric protein composed of 4 polypeptide chains, 2 alpha and 2 beta. Its efficiency and multifunctionality makes it a central focus in the study of respiratory physiology, biochemistry, and medical research. First of all, anemia, as you all are aware of this, that hematological condition characterized by deficiency in the number and quality of RBCs. Various outcomes like increased tiredness, reduced functionality, heart failure, these all are associated with anemia. Then we discuss its prevalence by means of two main groups, by demographic distribution and then by geographic regions. It is more common in the children and then in the women of reproductive age and then in pregnant women, and then by geographic region it is more common in the developed underdeveloped countries and later in developed countries. The method we used during this, first of all, what is molecular docking? Molecular docking is a method that performed orientation of ligands to receptors, which is useful for drug design and biochemical analysis. First of all is selection and retrieval of phytochemicals as ligands. List of phytochemicals were obtained from the IMPART and TCMSP databases, and almost 30 phytochemicals were identified as phytochemicals of beetroot. Then for the retrieval and preparation of receptor proteins, we obtained the 3D structures of our proteins, which were oxyhemoglobin, hemoglobin, and anticalin from protein database bank in PTBQ format. And later for the preparation of receptor proteins, we used a software MOE, molecular operating environment. Um, in this, we remove the extra bound ligands, extra water molecules, and introduce the new bonds in order to improve our structure of proteins. While during molecular docking, we use two basic tools, Pyrex and Discovery Studio, and then we identified the ligands according to the RMST value and their binding affinities. Pharmacokinetics parameters for drug scanning. During this, the admit profile and Lipinski rule were followed. What is admit profile? Um, those our ligands that follow the absorption, metabolism, and toxicity rules. While in Lipinski rules, we see the five main things, and that is molecular refractive index, molecular weight, hydrogen bond donor acceptor, and log p value. That should be less than 5. And next step is collection of plant material. Plant was collected from a local market in Faisalabad and then verified in clinical biochemistry lab at Government College University, Faisalabad. While in vitro preparation of the extract, in this, first of all, we chopped our sample into pieces and then dried and grind it into fine powder. Then, for the preparation of extract, we took the ratio of ethanol with sample of 1:10 and then placed on the shaker for 48 hours. Later, we transfer our sample into the apple drop and let it to evaporate into small amount. For HPLC, for phenolic component analysis, we transfer our sample to the high-tech lab where they used ferrous chloride as an agent to perform the research. Beetroot use survey and verification. Almost 4 months were required for our survey, as we know that RBC is required 4 months for their maturation. That's why our survey were from November 2023 to Feb 2024. Meteorological parameter assessment. Almost 10 participants were agreed for this experiment survey or survey, and their hematological parameters were assessed before and after the beetroot application. And for quantitative data analysis, we perform the student t-test and standard deviation and mean value test for our results evaluation. Next is our results and discussion. These are the some parameters, HPHCT parameters that were assessed before and after the beetroot treatment, and control group was taken as a sample. HPLC analysis. This is the peak. It might be some unclear. Peaks are showing some certain chromatograms like caffeic acid and gallic acid and chlorogenic acid, showing the peak at a certain area and height at a certain time. Then analysis of interaction. Pyrex and Discovery Studio were used for their ligand protein interaction. Drug candidates with highest binding affinities were anticipated, and using confirmation scores, Pyrex visually represent the extent to which the ligands occupied the target molecule binding pocket, and out of 30 phytochemicals, top 5 compounds were selected on the basis of their minimal binding score for each receptor protein, followed by the Lipinski rule. Next, these are the our selected phytochemicals with receptor proteins showing their binding scores. Interaction analysis of the target protein. Main three proteins were selected: oxyhemoglobin, hemoglobin, and anticalin protein. First of all, interaction analysis of betanidyne with oxyhemoglobin. Betanidyne showed the interaction with these highest the lowest the binding score, best the binding pocket. Amino acids of protein betanidyne showed the strong interaction with oxyhemoglobin at about minus 9.6 kilocalorie, and each ligand has specific amount of binding energy with the receptor protein, and we will understand these that lower the binding energy, highest the strongest affinity, and best the binding pocket. Next is chlorogenic acid, that is showing the binding affinity at almost minus 8.7 kilocalorie with our oxyhemoglobin proteins. And next is interaction analysis of hemoglobin. Hemoglobin with silibinin ligand. Silibinin is showing the best binding pocket with hemoglobin at about minus 9.4 kilocalorie, and later the vitamin E showing the interaction at about minus 8.6 kilocalorie. All of these ligands have different binding affinities with their receptor proteins. And last is the interaction analysis of anticalin protein. Silibinin showing the binding energy of minus 7.8 kilocalorie with the anticalin protein. And last, ascorbic acid was selected just because, although it has the lowest, highest binding energy, minus 4.7, but due to its it is not it was not selected because it's violated the Lipinski rule. We just selected the phytochemicals that violated just one rule out of all. uh, Later we will see in this that druggability analysis. The selected best phytochemical candidate must adhere to minimum of 4 rules out of the 5. Out of all of the leading substances, betanidyne, ascorbic acid, vitamin E, silibinin satisfied all the criteria specified by ROF, while alpha-spinstrol and chlorogenic acid broke one rule, and other ligand like sucrose violated more than one rule. That's why we rejected the sucrose and rutin as well. Next is drug-like properties of the best selected phytochemicals. These are the Lipinski rules that were followed by our ligands. And next is admit profiling for phytochemical against one oxyhemoglobin, hemoglobin, and anticalin. Uh, all of the chosen phytochemicals were determined to be non-carcinogenic. However, one phytochemical, namely betanidyne, were identified as being hazardous according to the AIM test. That's why we will not select betanidyne. The comprehensive drug profiling indicates that phytochemicals recommended or chosen in this study could potentially serve as a medication candidate. Next is our admit profiling, showing absorption, metabolism, and toxicity. Just betanidyne violated the toxicity rule, while others all were followed the same rule. Next is conclusion, that we will conclude our research with this, that the phytochemicals selected after the molecular docking were considered as a best drug candidate due to some reasons, like they follow the Lipinski rule, and admit profile was best. However, their lowest binding score with receptor proteins predicted best binding pockets, and they fulfilled pharmacokinetics and pharmacodynamics parameters. Moreover, after the survey of beetroot application, meteorological parameter assessment showed a direct, remarkable positive outcomes that actually proved that these phytochemicals are best drug candidate against anti-anemic activity. Okay, thank you. If any question, then you may ask, please.
Thank you so much, Javeria, for your presentation. We have some questions for you in the chat box if you might um, answer them. Until then, I would like to call upon our next presenter, which is Mahnur Shams from Federal Urdu University, Karachi, Pakistan. I would like you to start with your presentation, please, and I would again request you to please keep it short and concise. So, Mahnur, over to you. Mahnur, if you are here with us, please continue with your presentation.
Yeah, hello. Assalamu alaikum. Am I audible?
Walaikum assalam. Yes, Manu, you are
Hello.
clearly audible.
Okay. This is me, Mahanur Shams, oral presenter from Karachi, Pakistan, Federal Urdu University, Department of Microbiology, Laboratory of Infectious Diseases, Immunological Research. I'm a research student, and my research topic is on exploring the antimicrobial and antioxidant properties of metallic oxide, nanoparticle, includes zinc oxide and copper oxide. So I'm starting to sharing my screen. Please tell me that my screen is visible or not after I've shared it. Okay. My screen is visible. Hello.
Can you hear me, Manu? Your screen is visible. You can continue with your presentation.
Okay, thank you. So my research topic is about the exploring antimicrobial and antioxidant properties of metallic oxide nanoparticles, zinc oxide, and copper oxide. The reason I choose this topic because of the antimicrobial resistance, which is becoming the global issue nowadays, the antibiotic becoming the less effective. The reason behind it, the overuse and the misuse of the antibiotics. And according to the Center for Diseases Control and Prevention, multi-drug resistant bacteria causing over the 2 million infections. Like, this is the stopping point for us to think about it, like the bacteria, the multi-drug resistant bacteria causing the 2 million infection each year. So the pandemic and epidemic becomes so common due to this reason. So to overcome the antimicrobial resistance issue, I had choose the nanoparticle as a substitute of the antibiotics. The nanoparticles, basically a solid particle having a diameter between 10 to 100 nanometers. And especially metallic oxide I had choose because they are the most promising one as the antimicrobial agent. So, okay, moving towards the methodology. But before moving toward the methodology, I would like to tell that why I choose the copper oxide and zinc oxide is because they have the less complex structure, they are easy to prepare, and they are cost effective as compared to silver oxide and a gold oxide. I had prepared the nanoparticles by mixing the two different solutions, as you can see on the screen, by giving the specific heat and saturation. And after that, I had got the two different nanostructure, white product based obviously of zinc oxide and black product of copper oxide. You can see the XRD graph, X-ray direction graph, diffraction graph. Basically, the pinpointed peaks tell us about the crystalline size of the zinc oxide, 30 nanometers, and the 17 nanometer of copper oxide. They also it also tell us about the hexagonal vertex structure of the zinc oxide and monoclinic crystal structure of the copper oxide. I had done the antimicrobial activity by a very convenient method, a Gowel diffusion method, and I had chosen gentamicin and griseofulvin as standard drugs against the pathogenic strains. I had chosen the many critical gram-positive, gram-negative bacterial strains, and the fungal strains. Some of them are Proteus vulgarius, Staphylococcus aureus, and Candida albicans. A little part of my research is also about the antioxidant activity, which I had done by a very well-known method, DPPH method, and I had used ascorbic acid as a standard. Here you can see on the screen the scanning electron microscopy image of the zinc oxide and the copper oxide, which clearly tells us about the shape of the both nanoparticles, the rod-like formation of the zinc oxide and ovate-like formation of the copper oxide. These are the drizzle tables as zone of inhibitions of the copper oxide and zinc oxide against the gram-positive bacteria and gram-negative bacteria, which clearly tells about that copper is more better than zinc oxide. The major reason behind it, the copper has a less crystalline size than the zinc, which is the best point for copper because the less crystal size, the more great antimicrobial activity. Is the antifungal and antioxidant table charts. You can see again the copper oxide is comparative good than zinc oxide. And in antioxidant activity, the percent inhibition of copper oxide is 92.76, EC50 160.25, while zinc oxide is 70.15 EC50. And you can see the zone of inhibition in the picture. Okay, moving towards the conclusion. So it's concluded that the nanoparticles can be used as a substitute of the antibiotic because they have their unique properties. They have the less complex structure, not like antibiotic. Antibiotic have a huge structure while nanoparticle have a very less and small structure. They have the nanopores. They can easily penetrate into the microbial cell. They have ability to rupture the bacterial cell and can produce the oxidative stress against the fungal cells. This is the plus point of the nanoparticles, especially of the copper oxide and zinc oxide, as well as they had also proven them as a good antioxidant agent. So against the as comparative to the ascorbic acid. So my research proved that nanoparticles can be used as a substitute of the antibiotic. Here we can find a solution against the antimicrobial resistance. Thank you.
Thank you so much, Manur, for such a concise yet knowledgeable presentation. We literally loved your presentation. It was amazing. I would like to, if you have any questions, audience, please you can ask in the chat box so that Manur will be able to answer them. I would like to move on to our next speaker, that is Mohammad Amar from Capital University of Science and Technology, Pakistan. The virtual stage is yours, Amar. Continue with your presentation, please.
Thank you so much. Am I audible? Thank you so much. Am I audible?
Amar, you're audible, but your volume is really very low. You're hardly audible.
A second. Am I audible now? Am I audible? Can I start?
Yes, Amar, you're perfectly audible. Please start sharing your presentation with us.
Is my screen visible to all?
Yes, your screen is visible. We need to see your presentation.
Now it's visible to all. Can I
Yes,
start?
you're present. Yeah, you can start perfectly.
Thank you so much, World Forum for the Young Scientist, for providing me this opportunity to share my research on high consanguinity in Pakistan. My co-authors are Seher Safdar and Dr. Saeeda Maryam Bakhtiar, and we are from the Genetic Molecular and Epidemiology Research Group at the Capital University of Science and Technology. Due to time constraint, I will just start my presentation very fast. As you all know that human genome is 99% identical in every human, but 1% it's variable to person to a person. And there are few common definitions like when our DNA it change any nucleotide or any frame shift that we call it a mutation. But if a mutation is in the population and the 1% more than 1% of the population contain that specific mutation, we called it polymorphisms. And in the Pakistan, we believe that there are multiple polymorphism for the different diseases that are causing a huge genetic inherited genetic burden in the population. And the reason that is supporting that genetic burden is we believe that is cousin marriages. And the most common reason for the cousin marriages are our social or culture or economical and the religious reasons. Moving forward to the literature view, Pakistan is fifth largest population, and it's not coming lower because we have highest population growth rate that maybe in the future it will increase to the third most largest population in the world or the second. And despite addition to this, there is high level of consanguinity. A consanguinity or a cousin marriages is not limited to the first cousin. It include second cousin, third cousin, and the fourth cousins. It have multiple degrees, as I mentioned in the presentation. The fourth cousin degree, or when we called it in Urdu at that inter-biradari marriages. In the inter-biradari marriages, the coefficient of interbreeding is comparatively higher than the first and second cousin marriages due to the genetic makeup. So in the Pakistan, we are practicing cousin marriages at a very high rate. Uh, so coming toward the objective, our research was basically to identify the correlation between high consanguinity in the Pakistan and how it is one of the biggest public health threat in the Pakistan. What are the drawbacks? How the both things are linked. The major hindrance we have in our research is that in Pakistan there is not much published data on the genetic disorders prevalence. So we use multiple strategies to encounter that we will discuss later. In our methodology, first was the data collection. We collected the data for the 30 most common genetic disorders in the Pakistan from the literature as much as we can get, and 72 different countries data of the consanguinity, and then we applied the statistical methods, and later on we analyzed the data, and the visualization of data was done. These are the few formulas. We will not discuss this in detail. One is the Bayesian theorem, and second is the coefficient of interbreeding that we utilized in our methodology. Here are our results. The upper graph show the uh, at the bottom there are multiple countries, 72 different countries, and at the vertical axis we have the percentage of the consanguinity. Pakistan is fortunately having the most highest consanguinity rate in the world. We see in the Islamic countries the consanguinity rate is higher as compared to the non-Muslim regions, but in the Pakistan the rate is much more higher even than the Muslim countries. It's approximate 70 to 80%. And again, for the interbreeding coefficient, the Pakistan is also leading, followed by the Afghanistan, Saudi Arabia, Iraq, and these all are the Arabic and Muslim countries. Similarly, the graph on the left shows the percentage or prevalence of the genetic disorders in the Pakistan. These are the 32 genetic disorders. We found this prevalence by using the Bayesian theorem, in which we use the small population to guess the prevalence in the whole population. And the second graph on the right side shows that how much these 32 genetic disorders are contributing to the overall prevalence of those genetic disorders in the world. And this is enormous, and it is a direct relationship between the genetic disorders prevalence and the rate of consanguinity in the Pakistan. Moving towards the conclusion, our research is a link between high consanguinity and the genetic disorders, and we believe there are multiple cultural, religious, and social factors. We are divided into many groups. We speak more than 100 languages in Pakistan. And there are multiple social and cultural groups. We do not marry in sub-religious divisions. So that is the one thing that is leading toward the genetic disorders in Pakistan is called as the gold mine of geneticists. This is the major reason for that, and for this we recommend the nationwide initiative. We believe, according to the WHO, if the disease is disposed in the population, and the population, one more than 1% of the population have this genetic polymorphisms, there should be a screening program. And thalassemia is 7% in Pakistan, and there is no proper screening program for the thalassemia. So the national initiative for the prenatal diagnosis for the screening program are very much compulsory. And secondly, we need a lot of genetic counselors in Pakistan. There are currently only two licensed genetic counselors in Pakistan. There are few others that are practicing, but they are not licensed officially by the government of Pakistan. So we need a genetic counselors in Pakistan, especially in all the public hospitals, and there should be a designation of the genetic counselor at the division level of the Pakistan. This is our research group, Genetic Molecular and Epidemiology Research Group. There is my email. Thank you so much, everyone, for listening. If anyone have any questions, please feel free to ask, and we are always looking forward for the collaborations. You can contact me using this email. Thank you so much.
Thank you so much, Amar, for such an amazing presentation. Your presentation was not only visually very appealing, but your presentation skills are also very commendable. We look forward to have many more collaborations with you in future also. So if you have any more questions, you can ask Amar in the chat box. Moving on to the next presentation, we have Saifullah from University Faisalabad. Saifullah, we welcome you warmly on the WFYS platform. Please start with your presentation. The virtual stage is yours.
Assalamualaikum. My slide and my voice is visible to and audible to everyone.
Yes, sir, your slides are visible and you are perfectly audible.
Firstly, I would like to thank Muzammil Shazad and Dr. Sayyid Anza for inviting me and giving me such opportunity to discuss my research on the international level. I am Saifullah and PhD scholar in the University of Agriculture, Faisalabad. I just give a overview. Actually, my study is so complex. It is not easy to understand everyone. In my study, actually we use express sequence tag and predict the gene which we think anyone can this confirmed by the PCR. Firstly, my research is the in silico prediction of drought response gene in barley using express sequence tag. In introduction, as you know, due to climate change, there is a different type of stresses are increased, like biotech as well as biotech. We study the barley. In my study, barley is a model plant, but due to drought, it yield are also reduced. And is my slide again visible?
Saifullah, your slides are not visible as of yet. Please try sharing your screen once again. We can see your slides now. You can continue with your presentation.
Okay.
Saifullah, unfortunately, we cannot hear you. Please unmute your mic. I think we shall move on to the next presentation, and our next presenter is Fatima Shakil from Government University, Pakistan. Fatima, please start with your presentation. We are so eager to hear what you have uh, for us today.
Am I audible? Hello?
Yes, Fatima, we can hear you. You can start with your presentation, please.
My screen visible?
Yes, your screen is visible and you're clearly audible as well.
Am I slide visible? Are my slides visible?
Yes, yes, your slides are visible. Fatima, you can start with your presentation.
My name is Fatima Shakil. I'm from Government College University, Lahore. So first of all, I would like to thank your Korean scientists for giving this opportunity. My topic of research is the impact of research implementation of egg production, particularly in hatchery in Japanese food. ICALS is a supplementary way of given to Japanese food for Japanese breeding factor that egg production, fertility, and hatchery in Japanese food. So that's what I did. Let's start with the introduction. Starting with our introduction, the Chinese goal is the small bird which was used as a fermentation in 1900 and in mostly in Japan and in the East Asia, Japan, Korea, or even part of Russia and China. It was considered from 1900s from 1900s just because of its melodious voice and songs were also popular at that time. So its demand really surged at high in early 1940 due to its nutritional value and its delicacy and richness of taste. In Japan, it's got really popular. The most important thing is that it's for one of the physical research animals in zoology, just because of its small size and rapid reproductive cycle and straightforward care requirement, not such specific care requirements that Japanese will require. It's mature quickly, reaching sexual maturity in about 8 weeks or almost. The main topic of today was the role of vitamin E, selenium, and zinc, which is the supplement was given to the Japanese for protecting breeding or egg production that actually or fertility. So let's talk about the teams and objective of this study. So the first one, the most important one, it's to increase the egg production of Japanese forests to the egg production will lead to the more number of Japanese forests. So it is the most major factor. So now second, at the impact of vitamin E, selenium, and zinc on the egg production of catchability. And then the third will be the fertility of Japanese forests. So that's driving in methodology. Methodology means data by 16 Japanese soil and divided into controlled and experimental groups and male to female ratio of 1 ratio 3, means for one male and 3 females. The supplement was given only to experiment when the control was constant. So the first step was egg collection, and after egg collection, back to incubation and carrying. After egg collection, the eggs are placed into the incubator. Incubator with the temperature was kept to almost 90 Fahrenheit or Fahrenheit, which was only 65%, and the incubation period was 70. After incubation, I think they have actually incubation the gathering to check either whether the eggs were fertile or not. So fertile eggs were kept in the incubator and the unfertile ones were outside the incubator. After that, the hatched veggie players were taken to the group the area, but the box will for 500 chicken were captured. Based into the field of based knowledge management, and to that it will properly shift into the applications. There's a picture of Mark ORIC, and there is a picture of the and in result, we can clearly see that our supplement in the end production was here in an experimental group and environmental control. So it also in unfortunately we have significant difference in experimental group and control group. Also in attributability, we have seen major differences. So we have concluded that the that supplement will major and production tendency and attributability towards the Japanese gold grade country. So yeah, thank you.
Thank you so much, Fatma, for such a concise yet informative and knowledgeable presentation. We would love to collaborate with you in future also. I would like to give another chance to Saifullah because he dropped his presentation in the middle of the session. Saifullah, the virtual stage is yours. You can start with your presentation once again.
Sorry for inconvenience. Now my slide and I am audible to everyone.
That's completely okay, Saifullah. No worries. You can start with your presentation. We can see your slides and we can hear you too.
Yes, my topic is in silico prediction of drought responsive gene in barley using express sequence tag. In the intro, due to climate change, there is a different type of abiotic and biotic stresses are emerging nowadays. In our study, we select the barley. Barley actually as a model plant and it is a drought tolerant, but in harsh condition, it yield also reduce. The research question is that there is a lot of barley EST are already reported on the NCBI database and none of scientists study them. We use 6 steps. Firstly, we download through NCBI database, National Center for Biotechnology Information, then cleaning and confirmation, then transcription factor from these EST, then enzyme, and then their function and their alignment on the genome. After that, we predict the gene and confirm the gene by promoter study. This is the first step. We take 7008 EST and then Colleen as given in this table. We actually retrieve 1220 contig which are beneficial for our next study. Rest of the ESTs are not beneficial for us and it has a low GCL level and not to be used in the next study. And our third step is that after downloading, then cleaning, then contig formation. Actually, contig contain those sequences which are similar with each other, and this like as a batch or a group like. After that, one of the software which is called plant transcription factor. This predicts the transcription factor from these EST. From 1228 our contig, there is a 71 transcription factor are produced. The list of these transcription factor are given here. After that, we predict the enzyme which are coded by these EST contig. One of the software which is called Blast2Geo, which is used. This picture show that the maximum EST can take the code for the enzyme transferases followed by the hydrolysis and other are given also. After that, we align these EST on again by blast it on the NCBA to confirm that our downloaded EST are linked with barley or maybe from other species. But our study confirmed that the mostly of the barley species vulgar. After that, we check by gene ontology whether these EST are coded for which function. It may be a cellular process, biological process, and molecular function. After that, we by using one of the other database which is named KEG, which actually classify the our contig. On the other hand, what the metabolic process encoded for what. There is a different actually process which coded them. The table is given mostly involved in the metabolic process. After that, we align these our screen 1222 EST contig on the barley genome, check maximum similarity or alignment on the barley genome. After on the basis of the score range, we select top 10 EST contig for the next study. Then we align on the barley genome. There, this table show that to contig 10 it lie on the sixth position. On the other hand, other 9 contig also lie on the different position and different number. There is 37 position on which our contig are present. After that, we translate these contig by using this interpro second database to check these contig coded which protein. The name of the protein are given in this table. After that, we predict the gene structure. The structure of gene is given in this picture. Actually, the complete gene contain transcription start site, poly tail in mid. There is a first coding sequence, last coding sequence, and internal coding sequence. This is the actually complete gene structure. We also produce other gene structure which we can't select because they have no, some have no poly tail and some have no transcription start site. After the prediction of gene structure, we using one of the software which name is the phytosome. Actually, we confirm that by study the promoter of these gene to check whether cis regulatory element are present. These cis regulatory element are cross-checked by the literature. By checking that this table show these is regulatory element which is present in our gene predicted predicted ESTs in this picture. These cis regulatory element are already reported in the literature which show that they have maximum involvement in drought. This is the final conclusion. Throughout this study, we actually predict 10 those genes which contain more than 10 cis regulatory element which are already reported in the literature, which is beneficial for our study. Some of the future recommendation is that someone use the PCR and to confirm that they may be involved in the breeding program. And thank you. If anyone have question, you may ask.
Thank you so much, Saifullah, for such an impactful discussion on this very important topic. And thank you so much for your patience as well. Last presenter of this session is Selvin Burrito from Ireland. Selvin, please start with your presentation. First of all, thank you so much for waiting so patiently for your turn. You may start with your presentation, please.
Yeah, hi, can you hear my voice by any means?
Yes, you are perfectly audible and we can also see your slides.
All right, that's great. All right, my name is, hi everybody. It's a bit late on my timing. So my name is Selvin Barreto and I'm honored today to be presenting my presentation of artificial intelligence, the dynamics, the research efficiency, and how at this international conference in Life Science 2024. So my research focuses on the integration of artificial intelligence in microbiology to enhance diagnostic accuracy, improve laboratory efficiency, and predict antimicrobial resistant trends. So the basic what I'm going to do it right now is I'm just going to turn off my camera at the moment because I believe it might be lagging at the moment, and I'm just going to go on to my screen. All right. So basically, when starting up right now, just hold on a second. Yeah. So basically, starting with the first slide, I would say the first slide thing that I would say is regarding how the introduction will be artificial intelligence as a game changer in microbiology, wherein one thing for sure, artificial intelligence has brought up very great numerous technical uh, processes in this decade of technology of 2024. So first of all, I would say artificial intelligence has accelerated the diagnostic process, reducing time from days to hours, thereby enhancing the accuracy by reducing human error in microbiological identification. Third thing I would say, one thing which was very important is it offers scalable solution for high throughput environments like hospital and research laboratories, and thereby also facilitating real-time data analysis and decision making. Now, when it comes to the challenges with traditional methods, I would say one thing for sure, it's time-consuming manual techniques, where it's like more of a culture-based identification. Along with that, there are limited sensitivity in detecting certain pathogens, especially a polymicrobial infection, wherein there are multiple microbial infections at one time. And along with that, there's dependency on skilled technician, thereby it does uh, leads to an inconsistency. And also along with that, there is difficulty in handling large data volumes from dynamic genomic sequences. Now, why artificial intelligence is important right now? As I was watching the presentation by various presenters in and around Pakistan and in and around the globe, the one thing that was very consistent, very keen was antimicrobial resistance, which is like a threat globally. So, wherein we need to important thing about it is that artificial intelligence is important right now. It's because we need to we have to undergo any rapid responses in any time an outbreak situation occurs, like the recent COVID-19 pandemic. Along with that, increasing availability of high big data in genomic and microbial studies. And also the most important thing, which will be cost effectiveness in research limited settings for faster and better diagnosis. Now, when it comes to the background of artificial intelligence is based on four main concepts, which is the machine learning, deep learning, natural language processing, and predictive analysis. Now, the main part of artificial intelligence is the machine learning, but it's like a model that improves diagnostic precision through a data pattern recognition. When coming to the external part, which is like the deep learning, it's like a multi-layered neural network that like processes high, complex microbial images and diagnostic data. Now, when it comes to the natural language processing, what it depends upon the artificial systems from one system to another. The main concept of NLP is to analyze scientific literature to extract out the relevant microbial research. And one thing for sure, the fourth one will be predictive analysis, wherein artificial intelligence focuses trends in microbial resistance and disease outbreaks. Now, when it comes to the application, we have numerous amount of application including image-based diagnosis, wherein AI analyzes the digital images from microscope, thereby helping us identify whether it's a bacteria, a fungus, a virus, or a parasite. Along with that, it does help us understand what's a pathogenic genome, wherein it will identify whether it has any mutation, any resistance genes, and along with that, there's a clinical microbial automation, wherein I believe one of the colleges in the UK, the Middlesex University, has this system called as a MALDI, wherein it accelerates the microbial identification process. And along with that, the last one will be antimicrobial susceptibility testing, wherein it predicts the resistant pattern and thereby guiding the clinicians on appropriate treatment. Because we're using the traditional methods at the moment, it's fine, but in the near future, I would say it's just going to be really very hard to understand what is going on and how we're going to help whatever the resistant parts of a particular virus or a bacteria to be unknownly be identified as well. And now, as we speak about at the moment, I would say the recent advances is artificial intelligence has integrated systems and which has shown like 90 to around 95% accuracy in pathogenic identification. Thereby, along with that, it has performed genomic platform sequencing from data 50% more than the traditional methods. And along with that, AI-based AST tools significantly reduces the time required for resistant profiling. And along with that, that's one thing for sure. It does monitor the global microbial threats, which is very important. Now, coming to the objective, I would say the main. Sorry about this. Just a minute. Yeah, so the main objectives, what I would say is, I want to evaluate on the AI impact on improving the speed and accuracy of a microbial diagnosis. Along with that, determine the efficiency of AI tools in early detection and microbial infection. Along with that, assisting how artificial intelligence can help laboratories handle large databases and complex microbial patterns. And one thing, and another one will be understanding AI potential to predict how the antimicrobial resistance and the suggestive treatments. And my secondary objective is to compare the diagnostic accuracy with the traditional methods, explore the AI roles in genomic-based pathogen surveillance and outbreak management. And another will be investigate the AI efficiency in predicting microbial behavior and antimicrobial response, along with the examining AI use in automating data analysis and improving lab workflow efficiency. So the main methodology that I used was a study design, wherein a systemic approach was given to an AI application in microbiology from clinical trials to laboratory studies. And a mixed method research which I included was a qualitative and quantitative, wherein quantitative was a data analysis and qualitative was more like an interviews type to understand how and when it was done. Along with that, comparing the data analysis from a traditional to a diagnostic methods for bacterial, fungal, and viral. And along with that, understanding the case studies of AI-driven systems which are being used currently in clinical settings for pathogen detection. And the data sources which I used primarily was peer-reviewed journals focusing on microbiology and artificial intelligence that were published in the last 5 to 6 years. Along with that, understanding the MALDI and the PCR AI-based systems and image recognition systems as well. Along with that, clinical data from the hospital which are using artificial intelligence for microbial diagnosis, antimicrobial resistant tracking, and outbreak management as well. And another point was public data sets which included microbial sequencing, pathogen genome, and database as well. The inclusion criteria which I took was studies that have implemented AI for microbial diagnosis and microbial resistant detection. Along with that, peer-reviewed studies published helped me as a lot, and case studies wherein AI tools were traditionally compared with traditional diagnostic methods were used as well. The analysis method was mostly based on meta analysis of artificial intelligence performance, wherein checking out how the accuracy is comparing to the traditional methods, along with the evaluation of AI rules role in improving the clinical outcomes, and along with the treatment microbial infection. The statistic analysis of AI effectiveness in predicting antimicrobial resistance and reducing diagnostic errors was the method as one of them. Now, when I compared the data and I analyzed myself, uh, the speed and accuracy is the most important thing, which was very keen to me. Wherein AI tools reduce diagnostic time from around 50 to 60% in detecting pathogens in clinical samples. Along with that, the diagnostic accuracy improved as well, which was 90 to 95, as I said before, which with AI based, which was compared 85 to 90% in the previous methods. Wherein faster results led to a quicker clinical interpretation and improved patient outcomes. Along with that, AI systems were particularly effective in identifying rare and even hard to detect microbes. Now, the impact on clinical diagnosis was significant, was one of the which was significant improvement in identification of microbial infection. And along with that, one was AI-driven AST tools, which provided real-time data for appropriate antibiotics prescription. Along with that, it was integration of AI in clinical settings, thereby reducing the diagnostic error by around 25%. And one other point was AI enhanced laboratory workflow, resulting in around 13% increase in laboratory output. The third point was, which I noted down, was genomic application, wherein AI has improved, like sped up the genomic sequencing and interpreting the pathogenic genomes by around 40%. Along with that, improving the real-time tracking of antimicrobial mutation and resistant development. Along with that, it was one thing which helped was detecting emerging pathogens during outbreak more efficiently, wherein it was more of a endemic time, not but in when it comes to it will be like a pandemic, then it will help us in a lot in identifying and helping us prevent it to much greater cost than it would have been with this recent COVID-19. Along with that, there was a fast identification of resistant strains, thereby guiding the appropriate healthcare approach as well. Now, when it comes to as I said before, I would say that the benefits are ample as when it will be significant of time saving, improving patient management, enhancing the scalability of diagnosis, and along with that, the predictive model can help in anticipate any research trends as well. But the challenge is what is more important, because if there's a high dependency on high quality data sets to just train the AI systems, and along with that, there are sometimes bias between the training AI, and it can affect the diagnostic accuracy, especially in underrepresented population, wherein the population is not up to the maximum capability that is required for the artificial intelligence to work upon. And along with that, there is implementation of challenges, because if say for example a hospital is a resource limited setting, it cannot afford a high cost and technical expertise, because AI requires a large amount of resources, time, electricity, and high speed internet just to work upon it. And it is not possible on a small limited resources setting. And along with that, it requires continuous updatation and validation with new microbial data to just remain effective. And along with that, there was another point which was really concerning to me, was the ethical concerns, wherein the patient's data privacy is being used up, and the confidentiality if not is not given into consideration at all. Along with that, there was a transparency in AI decision making, which is often called as a black box problem. And along with that, there was a responsibility of diagnostic errors, and human oversight is still necessary despite the AI capabilities, just to check out whether it has done its work properly, or maybe it is just there for the sake of it is there. And along with that, there are chances if an artificial intelligence is being used, the the human the human which is supposed to be doing the job, there's a displacement because it replaces the manual diagnostic tasks in the laboratory as well. Now, I would say, the last conclusion of my takeaway will be artificial intelligence has dramatically improved the speed and accuracy, especially in high-end thought out settings. AI tools have essentially managed in predicting the antimicrobial resistance. Along with that, it has integrated the revolutionizing the pathogen tracking, thereby helping in containing outbreak faster. And continuous advancement in AI will further reduce diagnostic error for sure, and thereby improving the clinical outcome. Now, my future implementation is AI will be crucial in personalizing medicine, especially patients who have severe allergic reactions to certain antibiotics. When it will help us to identify what particular antibiotics is required for that, especially especially patients who have especially patients who are immunocompromised as well. An AI system will also help us to evolve in real-time integration with the hospital workflow, thereby staying persistent, which is very important in a high-working hospital as well. And further research is needed to address ethical concerns, which was data privacy, algorithmic transparency as well. And collaboration between AI developers and microbiologists is essential for refining the AI tools to suitable laboratory requirements. And thereby, I would just like to say thank you so much, and thanks a million to everyone listening to my presentation. Garamil Mohammed. Thank you.
Now screen is it so? Can you see the screen?
Yes.
That is good then. Okay, then I'm continuing my presentation. So I was telling that microbes are attracted towards the xenobiotics, and they form a monolayer on the top of these xenobiotics. Then they attract the other microbes, and in this way there is a formation of microcolony. Then they secrete a polymeric substance which is just like a glue material, and finally they form a microbial biofilm which then mature, detach, and this way it helps in spreading. But within this biofilm there are the microbial enzymes which are bound by the plasmids, and these enzymes are responsible for the degradation of these contaminants, including heavy metal as well as the dyes. So with all this background, the objective of my current work is number one, to isolate, characterize, and identify the metal resistance and dye mediating microbes from the polluted sites. Number two is to determine their biofilm formation by two ways: qualitatively via Congo red assay and quantitatively via CV staining assay. Number three is to determine the bioremediation potential of heavy metals, that is lead, nickel, manganese, zinc, copper, and cobalt in vitro. And finally, to decolorize the black liquor, which is a printing dye, which is used in paper printing like newspapers and all the assignments and the material we use in our offices. So that's black liquor. So we want to have a eco-friendly bioremediation of heavy metals, and this painting color using the microbial biofilm. So this is the graphic sketch of the methodology we followed. We first isolated dust grains from both the heavy metals contaminated as well as the dye polluted sites. Then like we isolated 16 microbes, and among those we screened for the biofilm formers, which are
positive for biofilm formation. Congo red is a red color dye. When the bacteria are positive for the biofilm formation, they produce black coloration. This is the qualitative detection of the biofilm formation. We also use CV staining assay for the quantitative determination of the biofilm formation. Then we characterized and identified those strains via 16S RNA gene sequencing who were positive for the biofilm formation. Next, we did the metal and the paper pulp effluent, which is the black liquor. Bioremediation, we treated this with the, we both treated the heavy metal as well as the black liquor with the bacterial biofilms. And then we determine the bioremediation efficiency using the atomic absorption spectroscopic analysis as well as the FTIR. And then we did the results interpretation and data analysis. So what we actually got. This is the slide which shows biofilm mediated bioremediation assay. This is atomic absorption spectroscopic spectrometry. So as I told you that we have in total 16 strains, and among those 16, only 11 were positive for the biofilm formation. We identified those strains via 16S RNA, morphologically, biochemically, and then we divided these strains into the various groups. So we made 7 groups, G1 to G7, on the basis of the mature biofilm. Then we used made 3 matters which we tested at a concentration of 100 microgram per ml, and then 3 matters at 2000. Because we did some preliminary analysis and we observed that the bacterial tolerance to these matters to up to this concentration. So we treated the bacterial biofilms at 100 for the manganese, nickel, and lead, and 2000 for the zinc, copper, and cobalt. So the test tubes were incubated with the bacterial biofilms having heavy metals for 15 days, 2 weeks, and then we did the atomic absorption spectrophotometry and FTIR analysis to observe any change in their bioremediation efficiency or the functional group, which is done, of course, by the FTIR. So for the black liquor, which is the printing dye used in the newspapers and so on, paper material. So we observed, we got 6 strains isolated from the black liquor contaminated sites, and we named them BC1 to BC6. And we added 3 milliliter of black liquor, and we treated this black liquor with these 6 strains for 3 time periods, that is 72, 120, and 168 hours. Following next, we checked the spectrophotometry, and we applied a formula to check the color removal by the bacterial biofilms. In this case, you can see this color removal percentage is, that is the absorbance of the uninoculated broth, and this is the residual broth divided by uninoculated broth multiplied by 100. So following next, we did the atomic absorption spectrophotometry as well as FTIR. What we got actually in results. So these are the Congo red results. You can see there is clear-cut black coloration of the strains on the Congo red, which are the red color pairs, which shows that these strains were the biofilm former. As I said, we have 11 biofilm formers among 16 in total. So this is the crystal wall staining assay, which is sort of quantitative determination of biofilm formation. We incubated the strains in test tube for 3, 5, and 7 days, and we observed that in total among our 11 strains, 5 strains, they produce maximum biofilm at day 5, while the remaining strains produce maximum biofilm formation at the day 7. So 2 groups of the strains at day 5. So we treated day 5 strains for the separate matters, and day 7 strains where mature biofilm formed for the separate with treatment with the separate matters. This is a brief morphological characterization of the isolated strains. This is very just like for you to have a look. I mean, we have all type of strains. Then this is the morpho of these total 11 strains total. And this is the chemical characterization. We did the fermentation test, we did the urease, catalase, MRBP, and so on. So on the basis of morphological and biochemical characterization, strains were identified up to only genus level. And we observed that strains might be Bacillus, Klebsiella, Lactobacillus, and Pseudomonas. While next we did the 16S RNA gene sequencing, and we observed that our strains actually belong to same, but up to species level we got like Pseudomonas putida, Bacillus lacaniformis, Lactobacillus saccharicus, Klebsiella pneumonia, and we constructed a phylogenetic using the type strains which we obtained from the NCBI website. Okay, so next we did the the bioremediation assay. This is the bioremediation of lead, nickel, and manganese, three metals, right? And we treated with the 7 groups of the bacterial biofilms. As you can see in this figure, that the highest bioremediation efficiency was done by G1, which is 67.4 microgram per ml, and this is for the lead, and this is done by G1. Next is the G3, which did the highest bioremediation efficiency of manganese, right? And highest degradation of nickel was observed by the G7. So different bacterial groups, because they have different preferences, they have shown different interest for the bioremediation of the metals. So these are the results by the atomic absorption spectrophotometry. And this is the bioremediation efficiency of the next three metals, which is zinc, copper, and cobalt. And we treated with these two group of the strains and then their combination. And we observed that L sphericus show the highest bioremediation of the zinc, while compared and their consortia when together, then there was much decline. But this is highest degradation of copper. And when bacillus species are alone, it again show the highest degradation of the copper. So this is the bioremediation efficiency of the color removal. That is the black liquor efficiency by the 6 strains. I told you that BC1 to BC6 were the strains which we obtained from the dye contaminated sites. And as you can see in this figure, the highest bioremediation efficiency was by P. putida, which removed this strain after 160 hours, because we treated for the 3-time intervals, right? And following next, K. pneumonia, which showed the highest bioremediation efficiency. Then there was like any format, and then the bacillus cereus, while B. subtilis and P. phlegmatitis showed the lowest bioremediation efficiency of the show the lowest color removal efficiency from the dye contaminated sites. This is the FTIR observation, because there was a change in the functional groups of the bacteria when they interact with the metals. So we observed the metals treatment before and after the treatment with the bacterial biofilm. For example, this is the lead control, and this is the FTIR. You can see that there are very amide, carboxyl, amino functional groups which are present at various types of the transmittance wave number. And when we treated it with the bacillus cereus, which was the SBS1, you can see that there was the observation of new functional group, like there was a shift in the peaks from the control versus treated. So this is the lead treatment with the bacterial biofilm. So next was the nickel. This is the FTIR of the nickel control. This is the absorption spectra, transmission spectra of the control. And when we treated it with a mixed consortia of biofilm, there was slight change in the functional groups of the which were present at the treated versus control. Like here is 1629, here is 1645. So there was a slight change in the peaks and the observation of the functional groups. This is the manganese, this is the control, and this is the manganese treated with the bacillus plexus. And again, there was a change in a slight change in because when the bacteria treat with the metals, then there was the change and treat the resistant metals, so the less resistance are made them available for the other microbes to detoxify them in an eco-friendly way. So this is the FTIR of the next metal, which is the FTIR of the liquid color, that is the black liquor, which is the printing dye. And this is their control without any treatment. And this is when it was treated with the bacterial biofilm, it was named as STN4A. And again, you can see that there was a change in the heavy observed functional groups. So this is the heavy metal treatment of, again, treated versus control from, because we have 6 metals, and then finally there are the conclusions. In this study, in total among 16, we have got 11 biofilm forming strains, which showed bioremediation potential of heavy metals and the black liquor. 16 sRNA gene sequencing showed Bacillus species as the dominant genus in the canaries and the pulp paper effluents. 6 isolates showed mature biofilm on day 5 are the others 5 showed mature biofilm on the day 7. Atomic absorption spectrophotometry showed lactobacillus sphaericus biofilm with the highest bioremediation potential against zinc. Among mixed culture biofilm, G7 showed better bioremediation against nickel and B plexus against zinc. So regarding BL decolorization, P. putida showed promising bioremediation potential. FTIR analysis showed that main functional groups, that is carboxyl, hydroxyl, carbonyl, as well as amide 1 and 2 are involved in the metal uptake. In general, there is formation and the displacement of many new peaks suggesting interaction of metals and the black liquor with the bacterial external structures. So future studies on the individual and the combined effect of the tested metal employing the understanding of biofilm cell communication, quorum sensing and modulating strategies will provide insight further into the mechanism of metal detoxification and bioremediation alongside the BL decolorization by the bacterial biofilm. So this is the publication we made with this research. And thank you very much for your patience. Do you have any questions? You are free to ask. Please feel free to welcome. And once again, I'm very much thankful to the organizers for giving me an opportunity to speak at this prestigious forum. So thank you very much. I'm looking forward to have any question.
A big thank you to you, Dr. Iram Liaquat, for joining us today and for sharing such a valuable knowledge with us. And your presentation was incredibly enlightening. We are also looking forward to collaborating with you in future also. So if you have any questions, please drop your questions in the comment section so that Dr. Iram might be able to answer them. Okay, I think I should be moving on to our next presentation, and it's my pleasure to announce Dr. Samina Shabir from the Women University, Pakistan, and she will be presenting on her topic and she'll be sharing her valuable knowledge with us. So over to you, Dr. Samina.
Thank you so much. Here is Dr. Samina Shabir. I am working as assistant professor in Department of Chemistry, the Women University, Pakistan. Today I am going to present. Let me share my screen first. Can you see my screen? Hello, can you see my screen?
Actually, we cannot see your screen. It's not visible to me. If anyone else is facing the same problem, please raise your hands. I cannot see the screen. Not visible to the audience as well.
Now check please again.
Yeah, we are waiting until the screen shows up.
Stop sharing because you have problems. There is a message. Sorry. So without screen, I can start.
I think yes.
So sorry.
It's okay. It's okay. No problem. No problem at all.
I think there is a problem on your side.
I think there is a problem with the meets because we were able to see the first few of the presentations, but after that it is collapsed. We cannot see any presentations any further. So let me look into the settings, and until then you can start with your presentation.
Okay. Can I start my presentation?
Yes, ma'am, you can start with your presentation.
Okay, thank you so much. Today I am going to present identification and characterization of protein couple receptor in melonfly and their expression analysis against beta cypermethrin. First of all, I will tell you what is GPCR protein receptor. G protein receptors are integral membrane protein containing an extracellular amino terminus, 7 transmembrane alpha helical domain, and an intracellular carboxy terminus. GPCRs are the largest and most diverse group of membrane receptors in eukaryotes. These cell surface receptors act like an inbox for messages in the form of light, energy, peptide, lipid, sugars, and protein. Such messages inform cell about the presence or absence of light, sustaining light or nutrients in their environment, and they convey information sent by other cells. GPCR play a role in an array of functions in organism body, and increased understanding of these receptors has greatly affected modern medicine. In fact, researchers estimate that between one-third and one-half of all marketed drugs act by binding to GPCR. Now, how GPCR receptor work? When any neurotransmitters norepinephrines are bind with GPCR receptor, and they activate the subunit of GPCR like GPCR receptor alpha, gamma, beta, and then they activate the adenine cyclase, and then they activate the cyclic adenosine monophosphate, and which further deactivates the protein kinase A, and ultimately increase protein phosphorylation. GPCR classification. GPCR divided into class A, rhodopsin-like receptor, class B, secretin-like receptor, class C, and class D, fungal mating receptor, and class E, cyclic nucleotide receptor, and class F, freezer-like receptor. GPCR function include neurotransmission, response to hormones, immune response, cellular organization, homeostasis, and sensitivity to light, smell, and taste. Here you can see the different scientists identify different GPCR receptor against different insecticides. We work in our lab in melonfly, and we identified GPCR genes in melonfly by genome-wide analysis. Melonfly, as you know, melonfly are most damaging pests reported from 18 countries lead to severe loss of cruciferous vegetables and rape seed crops. Estimated annual loss US dollar 0.77 billion per year. Global cost US 4 to 5 billions per year. Economic pest because of its ability rapidly to develop resistance to insecticide. We can control this pesticide by several methods like as cultural control, biological control, transgenic crops, and chemical control. But they have some negative impact on environments, and they are also affect the beneficial arthropods. Beta cypermethrin resistance, we are using here beta cypermethrin as an insecticide. Beta cypermethrin resistance mechanism study are ongoing in melonfly respectively, but there is no study available regarding GPCR receptor against melonfly. As we know that GPCR play an important role in insect physiology and behavior. Due to their specificity and importance, they have been considered as the target for new environmentally friendly insecticide. In this study, we did genome-wide identified the GPCR receptor in melonfly. Meanwhile, we also analyzed their expression pattern in response to beta cypermethrin exposure in different developmental stages and between different strains. Our study provides fundamental information that may further our understanding of GPCR signaling system in lepidopteran and aid in design of the insecticide or small molecule capable of disrupting the physiological process regulated by these signaling molecule and their receptors. Uh, uh, we use different techniques to identify GPCR. We use by blast. We use NCBI. Uh, we download GPCR. We download a genome from NCBI, and then we did blast by their relative genome, and we screen out GPCR, and then we prepare phylogenetic tree and structure domain analysis, motif analysis, promoter locations, and then we did some expression analysis. We identified 80 GPCR genes, which further subdivided into 4 GPCR subfamilies. 50 GPCR identified in family A and 17 GPCR identified in family B and 8 GPCR identified in family C and 5 GPCR identified in family F. And they have conserved domain. All of 80 putative GPCR had complete ORF sequence.
Dr. Samina, I'm so sorry to interrupt you in between. Can you please try sharing your screen once again because we have modified some settings?
Okay, sure.
Okay.
Can you see? Can you see, please?
Not yet.
Let me share again.
Okay.
Now.
Still not shared. Wait a second. Let me share my screen and let me know if you can see it.
Okay.
Let's just try it. Okay. Can you see it? Is it visible?
No.
No?
Yeah, now visible.
Is it visible now?
No. Sorry, no.
Okay. Yeah, still not visible. Okay, my screen is visible. The organizer has just told me. Can you see it?
What you share?
I just shared the conference schedule. Okay, someone in the chat box is also telling me that it's visible. Mariam is telling me that it's visible. Okay.
Let me share my screen.
Okay, let me stop sharing my screen and then you can start sharing. Okay. Okay, I've stopped sharing my screen and I want you to try sharing your screen again.
Yeah, now you can see. You can see my screen or not?
I cannot see it.
I don't know. Now,
Unfortunately, not. It's okay. You can continue. It's all right. You can continue.
okay. As I told you, we identified 50 GPCR that belong to family A. These 50 GPCR members of family A were identified in Malonfly and were further classified into three major subfamilies. Opsin, first is opsin that which include 5 genes, and biogenic amine receptor which include 5 genes, and neuropeptide and protein hormone receptor which include 40 genes. To uncover the detailed evolutionary and phylogenetic background between the Malonfly, B. mori, and Drosophila melanogaster protein employing multiple sequence alignment, and unrooted phylogenetic tree was generated. We developed 80 protective GPCR from Malonfly based on the genomic sequence. These 80 GPCR were divided into four GPCR subfamilies. Neuropeptide and protein hormone receptor. The largest subfamily of the rhodopsin-like families are the neuropeptides and protein hormone receptors. In current study, 40 protective Malonfly sequence were identified. Similar to other insect, Malonfly, rhodopsin-like neuropeptides and protein hormone receptors can be classified into different categories according to their ligands, allostrain A receptors, allostrain C receptors, and so on. Family B receptors are essential in numerous biological activities, including development, growth, and reproduction. They have extended N-terminal domains and link to a limited group of receptors and differ functionally and structurally from other kinds of GPCR. 18 GPCR from the Malonfly family B were identified and further subdivided into three subfamilies, B1, B2, and B3. Family C GPCR produce constitutive dimers with a wide exterior ligand binding domain. Family C GPCR contain three different types of GPCR: metatropic glutamate receptors and prides of SAMATLAS receptors and glutamate and aminobutyric acid receptors. Family F GPCR include frizzled and smoothened genes families respectively. And then we did domain structure of 80 GPCR genes, as you can see in this picture. Family A GPCR domain structure, family B GPCR domain structure, and family C and family F. They are all are different. And these all GPCR has different domains with respect to their families. Then we identified their exon, intron, and conserved motifs uh, uh, on the base of their GPCR families. These are showed that, as you can see in the pictures, the number of the exons and introns range from 27 to 1 and 0 to 26 respectively. Then the conserved motif in the 80 GPCR proteins were evaluated using uh, uh, by software. A total of 10 conserved motifs identified and conserved motifs shared by related proteins, as you can see in this picture. These motifs were identified based on sequence conservation because the function and structure of the GPCR are still unknown. Then we did expression analysis of these newly identified GPCR in response to beta cypermethrin. The current study used to qPCR to examine the expression levels of the 80 newly found GPCR genes in both susceptible and beta cypermethrin resistant strains of the Malonfly. The results showed that the expressions of the 50 mutated GPCR gene increased significantly in beta cypermethrin resistant strain, while the expressions of the 7 GPCR genes decreased significantly in resistant strain compared to control strain. Further, we choose 8 genes to check their expressions under LC30 and LC50. The LC30 value we use 143 mg per liters for LC30. Insecticides under beta cypermethrin insecticide employed to examine the GPCR gene expression regulation in melon fly. The finding implied that the potentially increased gene expression is associated with beta cypermethrin detoxification mechanism. As you can see in this picture, some genes are highly expressed. Maybe they increase this. Maybe these genes help in insect survival. Further, we can study on these genes. We can delete them and check their expression again. Conclusion. Summary of my results are these. The sequence genome of melonfly was used in current investigation to identify a total of 80 GPCR genes based on their conserved domain and function similarities. These 80 GPCR genes were further divided into 4 GPCR subfamily, 50 GPCR identified in family A, and 17 GPCR to identify family B, and 8 to family C, and 5 to family F. Melonfly GPCR showed significant difference in both the numbers of the genes in families and subfamilies as well as the sequencing of the genes. Further, we evaluated these 80 GPCR using RT-qPCR to confirm their expression in various stages of melonfly with insecticide or without insecticide. Interestingly, GPCR genes expression differently at different developmental stage and between different strains of melonfly. The most of the GPCR show low level of expression. Further, 8 highly expressed GPCR were selected to investigate their significant role against different beta cypermethrin doses. After 48 hours of the exposure to LC30 dose of the beta cypermethrin, the GPCR gene 67 and GPCR gene 68 and GPCR gene 66 and C1 and 71 and 25 and 42 were upregulated, and GPCR gene 67 showed a significant increase in expression after 12 hours of LC50 treatment, whereas GPCR 68 showed a significant increase in expression after 48 hours. Only a few highly expressed GPCR were identified, which may improve the organism resistance and would be effective insecticide target for melonfly management. Thank you so much. Is there any question?
Thank you so much, Dr. Samina, for such an engaging and informative presentation. Although we were not able to see it due to some technical issues, but your expertise were truly evident. And we are very grateful for your cooperation, your patience, and your contribution. And we are looking forward to collaborating with you in future also. So, you are most welcome. It's our pleasure. Dear audience, if you have any questions, please drop them down in the chat box. Until I call upon the next presenter. And all right, so it's time for our next presentation. So welcome with me, Dr. Mohammed Umar from Cairo University, Egypt, and he brings a wealth of knowledge on his topic. We are so eager to hear his presentation. Please take the word.
Thank you. Okay, thank you.
Dr. Mohammed Umar, if you are here with us. All right, so we are going to call upon our next presenter, which is Dr. Abrar Hussain from ICCBS University, Karachi, Pakistan. And we look forward to your insights, sir. Please take the virtual stage. Okay, so we just have, okay. All right. So, as per the organizer's instruction, I am okay. He will join later. He has just informed us he's going to join us later. Till then, we are going to hand over the conference, the presentation to Dr. Taibi Mariam, and she is from Ibn Tufayl University, Morocco. We welcome you warmly, ma'am, and the presentation is yours.
Hello, everyone. My name is Mariam Taibi. I'm from Morocco. I represent today Ibn Tufayl University from Morocco. And I will turn off my camera. That's because I have internet issues, and I will share with you my slides. Hope it's visible. Please tell me if
Is my presentation visible? Yes, we can see your presentation.
My name is Laiba Ishfaq and I'm from Center of Agriculture, Biochemistry and Biotechnology, University of Agriculture, Faisalabad. I'm here to represent the topic genome-wide identification of drought-responsive vicinal oxygen chelate, a glyoxylase 7, in response to artificially induced drought stress in Brassica species. Basically, Brassica belongs to Brassicaceae family and is one of the largest in the world, consisting of 435 genera and 3,675 species. It is closely related to model plant Arabidopsis. Agricultural plants that are included in this family are oilseed rape, canola, turnip, cabbage, mustard, broccoli, and cauliflower. Importance. Brassica plant have importance in agronomic benefits, medicinal benefits, breeding potential, research potential, health benefits, etc. Agronomic benefits includes they are hardy and resilient to various climatic conditions, so they can cope up with the food security challenges. Medicinal benefits includes they contain glucosinolates compounds that help protect against various types of cancer. Breeding potential, they have breeding potential related to disease resistance, climate resilience, and nutritional enhancement. They are widely used in research as their important model crops for genetic research due to economic importance, genetic diversity, and relatively simple genome structure. As they are low in calories and they are rich in vitamin A, C, K, minerals such as calcium and iron, fiber, and antioxidant. They're also a source of healthy unsaturated fats and omega-3 fatty acids. Economic as they are a source of edible oil and biodiesel, and they are widely cultivated for both food and oil production. The objective of the study were to characterize glyoxylase genes in Brassica species and comparative analysis of artificially induced drought stress in cultivated species of Brassica. First of all, the sequence was retrieved by using TIRE database. Then the sequence that was retrieved was BLAST through Hammer, and then BLAST-P analysis were done through BRAD. Then there was genomic characterization that include gene structure, information such as chromosome location, gene ID, start and stop position, and positive or negative strand. Then expression analysis were taken from the Brassica Naples transcript information resource. Then cis elements were determined and synteny analysis were done using the TB tools. Gene structural organization include exon and intron distribution. Then proteomic characterization include phylogenetic tree that was made by using BAGA 11 software. Subcellular localization was determined by using Wolf-P sort. And then motif analysis were done using the meme suit package. Physiochemical properties were determined using the ProdPram software that include length of amino acid, molecular weight, aliphatic index, isoelectric point, and instability index. This is the figure 1 represent the distribution of exon and intron. All glyoxylase genes in group 1, 2, 3, 4, and 5 contain 2 intron and 3 exon, except Group 5 that have members that contain 5 intron and 6 exon. Figure B represents the motif analysis that show that all glyoxylase protein contains 6 motifs, while 12 glyoxylase protein consist of 5 motifs. Only 2 glyoxylase oleracea and juntia member contain 8 motifs. The figure C represents the glyoxylase domain that was found in all the conserved motifs. The figure D represents the phylogenetic tree. It shows that there were 33 candidate glyoxylase genes. In Brassica carinata, there were 7. In Brassica juntia, there were 7. In Brassica oleracea, there were 3. And in Brassica napus, there were 8. And Brassica nigra had 4. These Brassica caseae glyoxylase genes were then characterized into 5 main groups according to their clustering patterns as shown. Figure 2 represent the cis-regulatory elements. Multiple abiotic stress and hormone-related cis-regulatory elements were identified. Most commonly, there were stress-responsive elements, especially MYB elements, MRE elements, ABRE elements, STRE elements, and DRE. It also includes Tata box, cat box, G box, I box, and W box, and hormone responsive element, etc. In figure 3, there were chromosomal localization and centering analysis. There were 12 chromosomes that were having different genes on them. The gene distribution was very stochastic, as BO3 contain more genes, whereas chromosome number 5 harbors only 5 genes, as subgenome 5 have only 5 genes. Each gene family has a representative on every chromosome, as shown here. The figure B represents the centering analysis that was done using the TB tool. There were 21 pair of collinearity genes between Brassica rapa and juntia, and 28 pairs were between Brassica napus and Brassica rapa, and only few genes were, such as 20, were between Brassica juntia and nigra. The predicted microRNA and 3D model structures. We determined 29 candidate miRNA from 9 families that specifically targeted the 12 glyoxylase genes. The miRNA 256A, B, C, and miRNA 165, F, J, and miRNA 317 targeted 3 genes, whereas miRNA 298B targeted only 9 genes, whereas MIR 5714 and miRNA 360 targeted only 1 gene. The figure B represented the 3D model structures of each of the 6 Brassica species. Protein interaction and subcellular localization. The protein interaction basically unveiled the glyoxylase protein query sequence. As the Brassica napus genome is widely distributed and it is available on the BRAD database. The result demonstrate that there were 9 functionally predicted proteins. And out of these 9 proteins, only 4 showed response to drought stress. The figure C here represents the subcellular localization. There were 33 candidates glyoxylase that were present in various subcellular compartments, such as nucleus, mitochondria, cytoplasm, nuclear membrane. Most commonly, they were positioned in the nucleus, and only 25 glyoxylase genes were present in the cytoplasm. whereas 32 proteins were predicted to be chloroplast localized, and only few were present in the mitochondria. And only one gene was translocated in the cytoplasm. The subcellular localization basically tell us that these genes are organelle specific and they express themselves under variety of environmental situations such as drought, osmotic, etc. The expression analysis. The expression analysis showed that the expression is high in certain tissue under drought conditions, particularly like in the leaves, in the petals, in the sepals and ciliate walls, but in the later developmental stages. So we say that these glycoxylase genes vary according different organs. They show distinct expression patterns. Whereas here the figure B represent that there were two conditions, drought, there were two conditions. One was control condition and the other was drought that was induced through the abscisic acid. Under drought condition, glyoxylase had minimal expression, whereas when ABA was induced, it showed significant effect in the roots, particularly at 3 or it gave higher value, while at 6 or interval it value decreased. So we say that the glyoxylase genes in the Brassica napus are mainly responsive to abscisic acid treatment, and they show more effect in the root tissues as compared to the leaves. Whereas here the figure C represent that the biochemical role of glyoxylase were checked under multiple environmental stresses. The gene here represent that in response to drought and osmotic stress, the publicly available data of Brassica napus that is available on BRAD database was determined in several tissues such as leaves and root at different time period. There were six, like they were 0 and 0 half, after half-hour interval, after 1 hour interval and 3 hour interval, the expression pattern was checked. Under control conditions, the napus gene show minimal expression in leaves as shown by the graph, and the expression was also low in the roots, while there was a gradual increase at 24-hour interval. How about the expression in roots show a notable peak at 6 hours, but it decreased under the control levels. Whereas in case of osmotic conditions, the expression in leaf increased significantly at 1 hour, and then its value was higher at 24-hour interval. This indicates that the gene is responsive to osmotic stress, and it showed higher expression in roots, weak value shown at 24-hour as compared to leaves. This is the morphological analysis. The 6 different Brassica species were given 3 peg concentration, 15, 30 and 45, and one was a control condition that was 0% peg. As we increase the peg concentration, all the morphological parameter decreased, and there was a significant effect when compared with the number of orthologs. As Brassica, Carinata and Juntia were having higher orthologs, so there was higher reduction in the growth pattern of morphological parameters when peg stress was induced. whereas under control condition, no significant effect, and whereas under control condition, they show higher growth rates. So it can be concluded that this study provides a framework for revealing the molecular mechanisms and role of drought stress in tolerance to Brassica species. By combining the transcriptomic, proteomic, and genomic data, we can cope up with the food security challenges, and we can develop such varieties that can cope up with the sustainable agriculture, and we can develop disease resistant varieties, and this can provide powerful tools for breeding potential. It's all up from my side. If you have any question, you can ask.
Thank you so much, Laiba, for such an amazing presentation. Without any further delay, I would like to call Umar Ali from Abdul Wali Khan University, Pakistan. Umar, if you are here with us, you can start with your presentation.
Is my screen visible?
Yes, your screen is clearly visible.
So let's start. Assalamu alaikum, and first of all, I would like to thank International Conference on Advances in Life Sciences for giving me this opportunity to present my presentation at this stage. My name is Umar Ali, and my topic of presentation is ecological dynamics of hornet predation on honeybee colonies and sites of sustainable beekeeping in Mardan, located in Pakistan. The topic is a topic from the entomology, which is related to the insects. Mainly, uh, two insects are focused here: Vespa species and the honeybees. Its predative behavior is focused in this study. Our presentation is categorized into the following categories, like first is introduction, then objectives, materials, results, and then last one is the conclusion. So in the intro section, we can see that beekeeping plays a critical role in sustaining agricultural productivity and ecological health. As we can see that honeybees are very important to for the pollination of many plants. Honeybees are very important for the pollination of many crops, like in the wild habitats or the domesticated plants. And in an area like Mardan, which is uh, vastly, there are a lot of plants and a lot of variety and diversity of plants. Uh, Furthermore, there are a lot of area for the uh, honeybees. So, a lot of threats and risks are present to honeybees, in which the Vespa orientalis or the Vespa species like Vespa carburu and Vespa tropica are the most probable one. Uh, in the next section, we have discussed our objectives, which include to hand out the interactions between Vespa orientalis, mainly focusing on the Vespa orientalis and Apis mellifera species. But still, there are some more related species like Tropica and Kaburu, which are mentioned here in this study. In the materials and methods, we recorded field observation through our senses and for capturing many type of video recordings and pictures were taken. Some chemicals were used for the preservation process. In Mardan, we have selected 4 areas, sub areas, and then we selected 20 afiris in these 4 areas. Up to 15 to 45 minutes, they were observed randomly for our. In the methods, we collected the data through our observations, and the arrived number of hornets and the bees caught were counted carefully. Whether the bees was alive or they were dead found in the firing. In the next sections, our results are shown in the form of graphs, which includes number of hornets arrived. Different like Vespa orientalis is present, Vespa carburo is present, Vespa tropica is present, and both the card bees alive and dead are shown in the figures. The first graph shows the data collected from the Kartlang area. The second is from the Shirgal area, and the third one is from the Tahtbai, and the fourth one is the Lunford, which are the 4 sub areas selected in Mardan. Our conclusion is that uh, uh, Vespa orientalis are showing more the Apis mellifera are generally honeybees colonies in Mardan areas and having a particular impact on those species of honeybees. They can cause to decline the honey production and can cause to minimize the colony size or can gradually lead to the colony collapse diseases and many more. Our finding also showed that large colonies were vulnerable to the hornet attacks, as many attacks and many successful attacks was found in the large colonies. In the recommendations, we recommend to protect the honeybees as possible. Location-based management can help to mitigate these predative activities of Vespa orientalis, or we can also adopt higher protection or can use repellent or can use different type of strategies uh, to um, to prohibit the visit of Vespa orientalis or other Vespa species to the Apis mellifera colonies. So thanks for your attention, and if there's any question, so you can ask.
Thank you so much, Umar, for such an insightful presentation. And we also thank you for your patience and for your presentation. Very quickly, I would like to move our next presenter, that is Ishfaq Yusuf from Nigeria. If you're here with us, kindly start with your presentation. And I would request again to keep it short and precise. Ishfaq, are you here with us? If you are here with us, start with your presentation, please. Okay, I think he is not here with us. So I am going to move on to our last international presenter, that is Selwyn Barreto from Nite University, Ireland. If you're here with us, kindly start with your presentation. We are so eager to hear your presentation.
Assalamu alaikum. Assalamu alaikum. This is Isaac Yusuf.
Walaikum assalam. Yes, Ishaq, you can start with your presentation, please.
Okay, thank you. Assalamu alaikum once again. I'm Isaac Yusuf from Aliko Dangote University of Science and Technology, Udil, from Nigeria. The title of my project is anti-diabetics and antioxidant effect of aqueous methanolic fruit extract 30 by 70% of velvet tamarind in streptozotocin induced diabetic rat. Hello, am I audible?
Yes, you are clearly audible, but we cannot see your presentation.
Okay, let me share my screen again. Is the screen visible now?
Not yet.
What is happening? Yeah, is the screen visible now?
Yes, we can see your screen now. Yes, your presentation is
Okay.
visible now.
Okay. I shall continue with the introduction. From the title of the project, as you can see, is anti-diabetics and antioxidant effect of aqueous methanolic fruit extract 30 by 70% of velvet tamarind in streptozotocin induced diabetic SARS. From introduction, firstly, you have to know what is diabetes mellitus. Diabetes mellitus is a metabolic disorder characterized by chronic hyperglycemia with disturbance of carbohydrate, fats, and protein metabolism, resulting from defect in insulin secretion, insulin action, or both. Diabetes causes long-term damages, dysfunction, and failure of various organ systems, leading to disability and premature death. Oxidative stress is the balance between cellular production of reactive oxygen species and the ability of cells to scavenge them. ROS are implicated in contributing to cancer, aging, inflation, and diabetes, etc. From the statement of the research problem, some toxicological studies regarding the use of synthetic antibiotics have shown their adverse effects and may also interact with some medication. Currently available therapies of diabetes are expensive and have a number of adverse effects. Therefore, the search for more effective and safer antioxidant and hypoglycemic agent, which will be inexpensive, highly available, closer to nature, and will be superior substitute of synthetic with no adverse effect, unless interfering with other medication is essential. Justification of the studies, the use of plant extracts and natural compound in the management of diabetics have gained increasing attention due to their potential anti-diabetic and antioxidant effect. One such natural product is velvet tamarind, that is Dalium guinis, a fruit commonly found in West Africa and Central Africa, which has been reported to have various medicinal properties, including anti-diabetic and antioxidant effects. The aims and objective of this study, the aims of this study was to investigate the potential anti-diabetic and antioxidant effect of the aqueous fruit extract of velvet tamarind in streptozotocin-induced diabetic SARS. And the objective of the study is to determine the body weight and blood glucose level of the streptozotocin-induced diabetic SARS. And the second objective is to evaluate the effect of velvet tamarind extract on serum insulin levels in streptozotocin-induced diabetic rat. The third objective is to investigate the effect of velvet tamarind extract on antioxidant enzymes activities in streptozotocin-induced diabetic class. So below or here are the methods of the project. Firstly, we have plant, that is plant extract, and identification of plant species. After we identify the plant species, then preparation, that is shed dried and grounded to powder of that velvet tamarind. Then we have to extract them, that is extraction. After extraction, we did acute toxicity, that is LD50 of that plant extract, which is velvet tamarind, and we measure the glucose level of that diabetic rat. And after we measure the glucose level, then we take the pancreas of that diabetic rat to lab for histopathology lab for histological studies. After that, we take the blood sample of that diabetic rat and normal and the other group of rat for insulin level, lipid profile, serum creatinine level, serum urea and electrolyte, then and total antioxidant, that is MDA and SOD. From the first method, that is acute toxicity, LD50. The method used to determine this acute toxicity was that described by Locke in 1983. The study was conducted in two phases. That is, in the first phase, we have three groups in the first phase, and we have 3 albino rat each in each group were administered with the extract at a respective oral dose of 10 milligram, 100 milligram, and 1000 milligram per kilogram body weight. The albino rat were observed for sign of toxicity and possible deaths for 24 hours, 72 hours, and 2 weeks. And in the second phase, we have 5 groups also of 1 albino rat each were administered with respective dose of 1250 milligram, 2000 milligram, and 2750 milligram, then 3750 milligram and 5000 milligram per kilogram body weight of the extract, which is velvet tamarind. They were equally monitored as in phase 1 for toxicity sign and death. Then that is induction of diabetes. In induction of diabetes, we have experimental disease diabetes was induced as described by Betten et al. In 2016, following overnight fasting, a single intraperitoneal injection of streptozotocin, that is 120 milligram of that streptozotocin per kilogram body weight, dissolved in a straight buffer 0.9%, was administered, followed by an oral administration of 5% glucose solution 2 hours after induction of streptozotocin. Then successful induction of diabetes was checked after 48 hours. Animal with blood glucose above 250 milligram per DL, that is per deciliter, were classified as diabetics, that is rat with diabetics. Then in our experimental design, the study was conducted on 25 albino rats. The rats were weighed and randomly assigned to 5 groups of treatment, and each group contained 5 rats, that is n is equal to 5 as follows. Group 1, we have diabetes-induced rat administered with only 200 milligram per kilogram body weight of aqueous methanolic extract of avocado, because we use 2 extract or 2 leaves. We have avocado seed and velvet tamarind. In group 2, we have diabetes-induced rat administered with 200 milligram per kilogram body weight of aqueous methanolic extract of velvet tamarind. In group 3, we have normal rat administered with only food and water. That is, we never administered it with that whether aqueous methanolic fruit extract of velvet tamarind or avocado or streptozotocin. On group 4 or on group 5, sorry, I have diabetes-induced rat administered with 350 milligram per kilogram body weight of glibenclamide. That is a remedy for diabetes. And we have determination of fasting blood glucose. Blood sample was collected via slight incision on the lateral tail vein using a scalpel blade. The initial fasting blood glucose was determined prior to induction of diabetes and recorded as initial fasting blood glucose 48 hours after inducing diabetes. The blood glucose was taken and recorded as a baseline, that is 0 hours blood glucose. Then the extract and standard drugs, that is glibenclamide, was administered to respective groups, and blood glucose was recorded 30 minutes, 2 hours, 4 hours, 8 hours, and 24 hours after administration using a glucometer, that is aqua-check GU. Then histological procedure. From histological procedure, pancreas of diabetes rat were removed, dissected, and washed immediately on ice called saline. A portion of this tissue was fixed in 10% natural formal saline fixative solution for histological.
All right, we are going to move on to our next presenter. Please keep it short. I am requesting you again and again because we do not have much time as all the other presenters are. Thank you so much for such an engaging and informative presentation. And your session was outstanding. So I would like you to, okay, thank you for sharing your screen. And if anyone has any questions, please drop them down in the chat box. Okay, so I suppose that I might be moving on to the next presentation. So So now we are ready to hear from Dr. Ashif Jahabar Ali. He is a professor in India, and we warmly welcome you, sir. Please start with your presentation. Sir, are you here with us?
Assalamu alaikum. May peace be upon with you. Assalamu alaikum warahmatullahi wabarakatuh. May peace be upon with you. Are you here? Excuse me, ma'am. Hello? Hello?
Yes, sir, you are audible. Please continue with your
Yes,
presentation.
okay. Yes, ma'am. Please invite the screen share my present. Optionable, please. Excuse me, ma'am. My screen is visible.
Yes, sir, your screen is visible.
Okay, wait. 1 minute.
Okay.
Okay. Shall we start? My screen is visible or not?
Yes, your screen and your presentation both are visible.
Okay. Thank you for giving opportunities. So this is from my name is Dr. Sheikh Jabar Ali from as a herbalist and assistant professor, Sedibadi Government Arts College from Tamil Nadu, India. So I can start my presentation. May peace be upon with you, first of all. My research topic, phytochemical screening, physicochemical parameters, and the evaluations of in vitro antioxidant activity on the aqueous extract of leaves of Lycos aspera from Siraikulam village of Ramanathapuram district of Tamil Nadu, India. So introduction, the plants, the God gift. So India is known as the equilibrium of medicinal plants because of availability of several thousands of medicinal plants in different bioclimatic zones. So in our India has the second largest biodiversity. So especially all the medicinal plants. So the all weeds contain the plants. So it's a medicinally properties. So I have chosen the locally available medicinal plants in Seraikulam village of the of Tamil Nadu. The plant profiles of the my our research topic. The plant profile is taxonomically so Lucas aspera. It's belong to the family of Lamiaceae members. So its vernacular name is Tamil name is Tamil name is Tumbai, and Hindi name is Goma Mutupati. Then description of the plant. Lycos aspera is an annual branched herb erecting the height of 15-60 centimeter with the short and have the actually granular stem and branches. So Lycos aspera, it's basically belong to the families of Lamiaceae members. It's contain volatile oil contains. It is leucans. The objectives of the studies, the objective of present work is a present phytochemical in vitro pharmacological activities and characterizations of aqueous extract. So why we have to choose aqueous extract? It is a universal solvent when compared to ethanol extract or chloroform or diethyl ether or methane, etc., etc. So these are the it is aqueous extract. It's a highly it is a universal solvent. So that's why we have chosen aqueous extract of leaves of Lycos aspera. The main objective of the present work is prepared crude extract with the aqueous solvent system, and then pharmacognostical standards like ash values, extractive values, and fluorescence analysis. Preliminary phytochemical screening of the plant extract, antioxidant activity of plant extract, and thin layer chromatography of the plant extract. So then next materials methods. Then first we go for preparation of plant material. The plant should be collected from Lycos aspera, were collected from Seraikulam village of Ramanathapuram district, Tamil Nadu, India. The orchard specimen is stored in Department of Botany, Sedibadi Government Arts College, Ramanathapuram. The collected plant were immediately brought to the laboratory using separate poly bag and dried. So then here we have to point out the how to collect the plant material, which time was suitable collected time. At the time of early mornings up to time of 6 a.m. to 8 a.m. So it is suitable for especially lamiaceae members of medicinal plants. It is it's applicable. So why? Because the plant after 12 p.m. or anything in noon time was collected from plant material. A volatile content may be evaporated easily. So that's why the plant material can dried under the sad conditioner. So we should not dry it under the direct sunlight. So because of the plant volatile compound, it is easily evaporated. So that's why we have to dry it under the sad conditions and then grind it for a stainless steel blinder with using a mixer, and then go for suitable pack Ziploc cover for further studies. So then after that, the stored powder material materials that be taken further work. So then we go for solvent extraction. The leaf of Leucas aspera were dried under the sand, mixed together, then they made in the two powder with the medicinal blender. Then go for solvent. We are using here. We are using Soxlate extractor. Then determination of fluorescent behavior of the plant border using following chemicals: H2SO4, HCl, HC3OH, and powder were derived in the normal daylight and under short wavelength and long wavelength. So short, then normal daylight, powder analysis would be taken from normal daylight, and then go for short wavelength to long wavelength. Then physicochemical content, that be loss and drying, determination of establish the method of procedure, and determination of moisture content, loss and drying. Then determination of total ash value, and ethanol soluble extractive and acid soluble extractive, water soluble extractive. We are following the standard procedure. Will be follows. Then powder will be the extracted. The extract will after that the extract will be tested. Phytochemical studies in the preliminary phytochemical analysis of whether presence or minus the qualitatively. So following phytochemicals, we are analyzed for the qualitatively. Test for alkalides, test for carbohydrates, and test for proteins, and test for steroids, and test for phenols, and test for tannins, and test for flavonoids, and test for glycosides, and test for terpenes, and test for gum and mucilages. These are the qualitatively whether the presence or absence in the qualitatively. And then go for a thin layer chromatography, TLC. Then whether we are finally the in vitro antioxidant activity and following procedure. First, DPPH radical scavenging activities and determination of total antioxidant active capacity, and superoxide anion scavenging activities assay. That's all. Then result. Coming to the result, fluorescence analysis. The result shows following chemicals in AlCl2, aluminum chloride, H2SO4, sulfuric acid, and HCl, hydrochloric acid, ammonia, and methanol. So these are the short wavelength to long wavelength. The following chemicals we can added in UV under UV visible chamber, and then determination of physical and chemical parameters like moisture content 7.19 and percentage, extractive values, ethanol acid extractive values, and 31%, and acid value in solid acid parameters and 87%, and ash values total ash value content of this plant 39%. So in preliminary phytochemical analysis following plants, for example, it's alkaloids present, gum and mucilages is absent, and glycosides absent, and flavonoids, flavonoids present, phenols present, and steroids absent, and sterol absent, and tannins present, and terpenoids absent, carbohydrates present, and protein absent. So these are the preliminary phytochemical studies we are analyzing. And then thin layer chromatography, TLC, and the spots can be totally 5 spots in we are gathering. Then antioxidant activities, DPPH radical scavenging activities. Then the plant should be increasing the compared with the standard ascorbic acids to Lycos aspera extract. So on the percentage of inhibitions, both 200 ml to 800 ml in per ml. So decreasing to increasing level. The DPPH dividing compared to standard ascorbic acids, DPPH is scavenging activities, it increasing percentage level. And total antioxidant activities same like that, standard with ascorbic acids, so decreasing to increasing order. Then superoxide anion scavenging activities, same like that, decreasing to increasing order. So then summary of this research work. So traditional Indian medicinal plants they've been used in the treatment of different disease in the country for centuries. So it is a locally available medicinal plants. It is a the Lycos aspera is mainly used to the local people. The leaf can be and leaf and the flowers. It's a flower is a white color. When added to mixed with milk and honey, it cured antipyretic. It's very good for antipyretic activity to cure some fever in traditionally. Till now, the local people are used in Tamil Nadu or local available area. Then these are the antipyretic activities. So antioxidant activities will be increased more when compared to decreasing to increasing. So it is where furtherly we go for cancer research. So these are the summary of this research work. So thank you for giving opportunities. Thank you very much. Any questions, any concern, any? Hello, ma'am.
Thank you so much, sir, for your deep knowledge and making this session truly impactful. And thank you so much for joining us from Tamil Nadu, India. We look forward to collaborating with you in future also. And if you have any questions, please drop them in the chat box so that he might be able to answer it. Till then, I am going to move forward to our next presenter, and that is Professor Kiran Rafiq from Jinnah Sindh Medical University, Pakistan. We warmly welcome you on this platform, ma'am. Over to you.
Assalamu alaikum. Thank you so much for inviting me on such a platform for sharing my research. And here I'm sharing the screen. Are you hearing me? Are you getting me? Hello?
It's wrong.
Hello, are you getting me?
Yes, we can see your slide now.
Yes, ma'am, you are clearly audible.
Okay, okay. Thank you so much. So my name is Kiran Rafiq. I am professor and the head of the department pharmaceutical chemistry. at Jinnah Medical University. And today I am sharing my research. And before starting the research, I would like to share something with you. That is, this is the fact that when we keep ourselves near to the nature, we away from various disease and disorders. In ancient times, it so happened that people used to use the utensils of metals uh, or soil and other things, but they did not use the utensils made from the plastics. But in the era of today, we are widely using the utensils made of plastic, and the plastic is very hazardous. My research is about that. The topic of my research is the study about the presence of phthalates in plastic materials, a precarious substance for human health. So first, let me introduce that what are phthalates. They are plastic owing to its effectiveness and durability. Is a material of choice for packaging as well as for manufacturing various products. It has widely used in daily life to boost the mechanical strength and improve the physical aspect of plastic. Various additives like antioxidants, flame retardants, and plasticizers are generally used. Phthalates are
Dr. Kiran, I am so sorry to interrupt you, but we are unable to see your presentation.
Okay, let me check.
Okay.
Now you can see?
Yes, ma'am. Now it is visible.
Okay, thank you so much. So phthalates, yes. So that plastic containing phthalates are precarious for human health, and they have tendency to leach into food and water from plastic containers, and they may disrupt in the endocrine hormonal systems. Endocrine disruption has been linked to reproductive issues, obesity, diabetes, and some cancers. It can convert into microplastics, small plastic particles that can be frequently ingested through food and water, and that may cause oxidative stress, inflammation, cellular damage, and consequently long-term health effects. There are high chances of phthalates to interact with immune cell, potentially impairing immune system. Phthalates mimic hormones and disrupt normal hormonal balance, leading to issue with fertility, fetal development, and reproductive health, and can also be responsible for increasing risk of birth defects. Ingestion of phthalates may cause gastrointestinal issues such as inflammation and gut barrier dysfunction, potentially leading to nutrient absorption problems. Commonly used phthalates in the plastic are di-2-ethyl hexyl phthalate (DEHP), diethyl phthalate (DEP), dibutyl phthalate (DPP), benzyl butyl phthalate, and dioctyl phthalate. These are the kinds of phthalates that are widely used in the manufacturing of plastic that are using in the utensils and other packaging materials. Plastics are accountable for pollution. As general issue, plastic pollution has become one of the major problems for environmental degradation. Waste accumulating from use of plastic is not properly managed. The methodology of my research that was adopted, it is based on the GCMS, and for that purpose, different types of plastic samples were taken as water bottles, expanded polyester, plastic spoons, plastic cold drink bottles, plastic container, and they were cut into 1 gram pieces. Each sample was taken and extracted by 15 minutes of sonication. And after that, 1 ppm and 0.1 ppm of the sample extracted was passed through filter into an auto sampler vial for analysis. The standard was also used, and that was purchased from the Sigma Aldrich. Instrumental conditions are mentioned here. The instrument was a Shimadzu GC2030 gas chromatogram. The GC was fitted with a splitter injection, port capped at 280 degrees centigrade. The Perkin-Elmer Colorus 600T mass spectrophotometer was appointed in full scan mode, and these are the instrumental conditions that were used. And after that, the analysis was done, and 5 different samples of plastic, including plastic cold drink bottles, expanded polystyrene, water bottles, plastic container, and spoons, were analyzed for the presence of phthalates. The gas chromogram of standard showed the retention time of DEHP at 14.705 minutes. All 5 samples showed presence of DEHP at retention time 14.798 minutes. Results.
Ma'am, I am sorry to disturb you once again. Your slides are not visible.
Okay, I check it again. Is that visible?
Yes, ma'am. Now it's visible.
Okay. The presence of 6 different types of phthalates was investigated in plastic utensils, which include the dimethyl phthalate, phthalate acid, monoethyl ester, di-2-ethyl hexyl phthalate, and benzyl butyl phthalate. And 1,2-benzene dicarboxylic acid and dicyl-octyl esters. These are the phthalates that were isolated from the plastic samples. So these are the results. These are the peaks of results. The analysis of 6 phthalate standards was performed by GC and MS, and then they were compared with plastic samples obtained from water bottles and plastic containers which are used in daily basis. The study concluded that phthalates were identified in various plastic materials, raising significant health and environmental concerns. So, phthalates can enter the human body and are associated with various health risks and toxicity and other developmental issues. Given their widespread use in consumer products, packaging, and medical devices, the findings highlights an urgent need for strict regulation, the development of safer alternatives, and increased awareness of phthalates' potentially effects on human health and environmental safety. These are the references.
وإذا كنت خرجت باللي يعني رجعت للدار ثاني صبري
Question, if anybody want to ask.
صبري نمشي للطواليت ونمشي.
This is all from my side. Hello.
Hello. Thank you so much. Please, I request all the distinguished guests to please mute your mics.
Okay. Good afternoon to you all. My name is Ashuru Ali Zain Al Abidin, a student of MSc Biochemistry from School of Science and Humanity, SR University, Warangal, India. Thank you for the opportunity. Thank you for to the organizers for this international conference on advances in life science and life science. Thank you for the opportunity to present my research work. My research work is phytochemical profiling, in vitro antioxidant activity, in silico antibacterial analysis of ethyl acetate leaf ester of Laniella oliveri. Introduction. Plants are a good source of biological active compounds. As we all know that plants are good active compounds, not as have a good source of biological active compounds known as phytochemicals, and these phytochemicals have been found to act as an antioxidant by scavenging free radicals and it also have some therapeutic values. Then we come up that the reason why we suggest this research, like in our place, like in Nigeria, because I am student, international student at ISR University. There is so many claims about this plant. Daniela Oliveri has some medicinal effect and medicinal value. Then we try to evaluate the phytochemicals that are present and test the in vitro antioxidant analysis, and also to test the in silico antibacterial analysis of that leaf extract of Daniela oliveri. Then this is the aim of our story. Then the sample collections, we collect the sample from Garin Ali Garko local government, Kano State, Nigeria, and the sample were identified and authenticated period to analysis at herbarium sections of Biological Science Department, Bayero University, Kano, Nigeria, and they give us a voucher number. Then after that, the fresh leaves were washed and subjected to shed drying for about 2 weeks. Then the dry leaves we grounded into them. Hello? Am I audible? Sorry.
Yes, sir, you are clearly audible.
Okay. Then the dry leaves were grounded into fine powder with a metal mortar and pestle, which is then stored in airtight. Then after that, we subjected the dry particle into substrate extraction. Then the solvent that we have used is the ethyl acetate. We use ethyl acetate to extract the phytochemicals. Then the phytochemicals, we check for the quantitative analysis. We check by phytochemical analysis. We check for the anthraquinones, polyphenols, tannins, flavonoid, saponins, cardiac glycoside, anthocyanin, terpenoid, phenols, and alkaloids. And the quantitative phytochemical screening, we check for the flavonoid and the alkaloid, saponin, and rest. Then for the antioxidant, we check for the total flavonoids, total phenolics, and total antioxidant activity, that is DPPH. And we check for the reducing power and hydroxyl radical scavenging assay. Then all of this, they have associated with this is the flow. And after that, the analysis, then we take the in vitro, in silico antibacterial analysis, where we get the data collection. Then we use PubChem database to get the natural product that are present in the Daniela oliveri. After we get the natural product after we get the natural product that are present in Daniela oliveri, that is through in silico analysis. Then we go for the ATMET analysis, that is the drug likeness of that natural product. After that, then we go for antibacterial activity prediction, where we use antibacter prediction server. Then this is the result of our study. We found out that for the qualitative phytochemical content of ethyl acetate extract, we found out that there is alkaloid is present, which is good for so many medicinal value. Polyphenoid, flavonoid are present, which are the very good antioxidant. Tannins are present, saponins are present, cardiac glycosides are present, then anthraquinones are absent. Then these are the results, where it shows that this plant is very good medicinal. And for the quantitative analysis, we found out that we have the highest number of alkaloid. We have a highest number of phenols, followed by alkaloids, then followed by the flavonoids, then followed by the tannins, followed by the cardiac glycoside, then followed by the saponins. Then for the phytochemical, for the antioxidant analysis, this is the result of the phytochemical analysis, whereby the percentage of inhibition, this part it's so close, but the control and the samples, this part there is so many variations where we cannot say much about that thing, since this one, the control has 93% of inhibition, whereby our sample has 68%. But despite the change in the percentage that we use, that is concentration, the ranges is not that too far, where we can also say by that thing it is very good antioxidant, because despite the we have the lowest concentration is almost within the range with the highest concentration. Then for the total antioxidant capacity, that is we get 5.0, and the control we use, whereby it shows that our control is less efficient as our sample. And also for hydrogen peroxide, then also our sample shows very good. And also for the hydroxyl radical, also our sample shows good inhibition. And also for this reducing power, our sample is almost, despite there is some difference, but our sample shows that it is good for the reducing power at different concentration. Then after that, the other result is for the in silico antibacterial analysis, whereby after we get the smells of our, we have about like 510 natural products that are present in the Daniela oliveri. Then we use admet Saba, so that we want to screen the ligands that have capacity to be drugs. That is the drug likeness of the ligands present in that plant, whereby out of that natural product that are present, we have five ligands by Led-Lipinski rule, and almost all the ligands are break the only 5 ligands that we suggested we take them for the further analysis. After we take our ligands for the further analysis, that we use that ligands to check for the antibacterial predictions. After that, we found out that this part, the ligands shows so many activity to so many microbacteria, but this part that we this result shows us the first line show the name of the bacteria, and the second line show that the ligands, that is the pest bacteria, Mycobacterium tuberculosis. Out of the 5 ligands that we have suggested, 3 ligands show inhibition to Mycobacterial tuberculosis, while 3 ligands show activity. Out of that ligands show activity on this, it is like this, and 4 ligands show activity on this rare pseudomallea and the rest. Then conclusion. This research revealed the presence of important phytochemical of high therapeutic value due to the presence of this phytochemical and other active compounds in the ethyl acetate leaf crude extract of Daniella oliveri. The plant shows some antioxidant activity which support its status as a very important herbal medicinal plant. The wide bioactive activities of the Daniella oliveri, which has been reported by different researchers, may be attributable to the presence of these important phytochemicals that we have shown, and our result also confirm that. And also the in silico antibacterial analysis reveals the inhibitory activity against mycobacterial tuberculosis, pistis, staphylococcus aureus, and aureus purpura, and the rest. Thank you for the listening. Thank you for the listening, and thank you for the opportunity. One has a questions or suggestion or anything else?
Thank you so much, Selvin, for such an amazing presentation. Your presentation was so beautiful, and so was your presentation skills. So as we approach the end of this remarkable conference, I cannot be adequately thankful to all of you for your participation, for your insights, and the enthusiasm you showed all the way through this beautiful session. And I hope that the connections we made, the ideas we shared, and the discussions that sparked will continue to inspire and drive our positive change in our perspective fields, in your perspective fields as well as ours. So all the suggestions that have been put forward during this session are being considered, and we will take them into account. Thank you so much, Muzammil Shehzad, for organizing this impactful session. And I would like to say to all of you to please stay connected with the World Forum for Young Scientists. And I look forward to seeing many of you again in the future events, sessions, and webinars. So this is your session chair, Sayyida Anza Hasnain, signing off. Goodbye.